Welcome to the session on viticulture, grapevine research in desert environment, and on the background of climate change. And why we do this conference here in Sde Boker. Yes, you don't believe it, but you are in Sde Boker. Mainly because 2,300 years ago, in this very area, they were actually producing grapes for wine making. Winemaking got into the region 6,000 years ago. And 2,400 years ago, Nabateans and later Byzantines were doing some good market out of wine production. The wine production was eventually distributed across the Mediterranean basin. Now we lost all that experience. Obviously the wine was different. We lost all that experience and we had a leg of production of wine, of viticulture for about 1,500 years. And with the state of Israel, everything started again, viticulture, mainly in the Mediterranean area of Israel. And today the Negev Desert is becoming front player in the wine industry in Israel. But it is also important that we talk about desert viticulture or viticulture in the desert as a model for viticulture in a changing climate. The desert can become a lab in the field where you can control irrigation to the drop. You can control input of fertilizer very, very accurately. You can control radiation. And therefore, the desert can actually be a model for what viticulture could look like in the Mediterranean basin and Mediterranean-like areas in the coming future, if the degrees, 2 degrees, the famous 2 degrees of temperature are going to actually increase worldwide. So welcome again. Now I'll give the word to Nurit, which worked out with me this fantastic list of speakers that will talk in the session, and enjoy.
So hello everybody, this is very exciting. I keep an eye on the number of participants, and clearly there is interest in this session. We are going to dive this afternoon or morning or whatever the time is, wherever you are, into 2 consecutive sessions, all dealing with viticulture, different aspects of viticulture in Mediterranean and arid climates. And we will hear lectures from people working on a wide range of topics related to viticulture. And the one thing that will tie between all of them is the relation between viticulture and environment. And with that, I think we will be happy to start with our first speaker, who, wait, I need to, I practiced. So Stefano Poni from Università Cattolica del Sacro Cuore from Italy. Aaron, don't laugh. So Stefano, please, the floor is yours.
Microphone, Stefano.
Okay, you hear me well?
Yes.
Okay, let me share the screen. Okay, do you see that? Do you see the screen?
Yes.
Yes,
Will do.
yes.
Okay, okay.
Yes,
Well,
yes.
thanks. Thanks for this invitation. Well, it's fairly unusual to be everybody online, but this is the way it is. So I have a short presentation on a case of drought resilience in temperate climate vineyard ecosystems, because as you can understand, if we look at the position of our viticulture area, you see this is the Po River, and we are here. We're basically in the middle of the Po Valley, so it's not a desertic area. It's an area where we get, you know, usual some rain in the summer, considerable rain, but we have a problem, and this is a problem that I would say 1 year every 2 we get into this. This is a Barbera vine that doesn't look very healthy. This is a sunburn damage on a wide variety. And this is the main river actually crossing town. That is basically with no water. So this is of course an issue. And so we need to handle this huge impact of climate change. Have you ever seen in your life an ugly vineyard like this one? I don't think so, but this is a Barbera vineyard in a year 2002 where we had a lot of rainfall. And you see, this is actually down in mildew that was not able to be controlled. And basically the canopy is defoliated. That was too wet. Well, if you look at the next year, this is how basically the same row would look like. Just to give you an idea of the variability in climate, that is the main issue. Because, I mean, anytime we start a new season, we don't know what's going to happen. It can be either very wet or extremely dry. And so we have been working over the past years on different factors and trying to look at the, effects of canopy geometry, training systems, orientation. We are now really into the cover crop versus tillage comparison. And then we have some new data about new rootstocks and calorie. Of course, I just have the time to give you just very short take-home messages. But I think that although it seems to be a sort of old topic, but it is not that old, because I think we need to have more information about the water use due to different canopy geometries. And I mean, we know basically that, either we have an overhead system or a DSP. Basically, we know that we have a linear relationship between the total intercepted light and the transpired water, okay? But the problem is, do we know exactly what it happens when the geometry of the canopy would change? Here we have cases of a goblet trellis or a GDC or a lyre system or a DSP. I'm not sure that we have enough information in general, although there has been a lot of work done on that. Let me give you the first example. Well, by the way, some of the data we'll be showing would rely upon our whole canopy gas exchange system, because, when you have a very complex canopy and you need to express the whole vine behavior, playing with the single leaf portable equipment is not very rewarding in many cases. And so we have been using this kind of equipment for a long time. And this is a kind of result that we get that is quite interesting to me. For instance, this is the diurnal trend of transpiration as the blue line and photosynthesis for a goblet trellis. So basically a round shaped canopy. And it is interesting noticing that basically at midday, you have maximum transpiration and maximum photosynthesis, but that is the time of the day where you have the lowest water use efficiency. Well, because of course, the VPD would actually drag this response and light would actually drag this response. And having a round canopy, the canopy that would intercept light does not change very much during the day. And, this is a sort of information that we should have for other canopy types. as well as for orientation. You know, the model would say that this is the way it goes. I mean, trying to model the light interception during the day for east-west versus north-south, but we also have real measurements on that. And it is very much confirmed that if you, at our latitude, so it's about 43 degrees, if we have an east-west orientation, of course, like in this case, we of course have a higher water use during the day, and we can estimate the saving of water using a north-south being the area that is actually enclosed here. I think anytime we plant a vineyard, we should take into account this kind of effects. Well, the other big issue that we are trying to face is the role of cover crops. Everyone loves cover crops, but again, the kind of information we have in terms of water use and competition towards the grapevine root system, it's not enough. Well, the soil management need to balance between tillage, herbicides, mulching, cover crops. But for instance, do we have enough information about this? This is a very nice example, an old study that was made in upstate New York. This is a soil volume, row axis, so depth and width, total herbicides. The yellow dots are the grapevine roots. So you see they're most shallow roots, right? But when you have at a given point in the alley, you have a cover crop, you see that when the grapevine root meets the cover crop, that tends to go below. Again, I think we would like to have really more information about this kind of interaction. For instance, you know, what is the water use of a cover crop before and after slashing? And what is the water use of a bare soil? We have been using also this kind of neat portable open gas exchange system to measure the water use by a given surface of a given water crop. And an interesting result was that we developed this kind of relationship. This is an exponential relationship that would actually relate the weight of the dry clip biomass versus the reduction in the evapotranspiration of the cover crop. That gives us some numbers that we can use to understand how much we can decrease the cover crop water use depending on the trimming that we made. And we are now very busy in trying to understand the behavior of new cover crop species. Here we have 17 new ones, and we also have built this handy, I mean, it's a small custom-made gas exchange system that is actually able to be moved quickly from one pot to another in order to get rapid measurements. And also we have set up other experiments in split roots where we have the cover crop growing in one pot and the other one is tilled, and with all the different combinations. Well, the good point for us is that finally it looks like that we might have some good new rootstocks for actually getting some tolerance to water stress. The most promising genotype is M4. We have a number of new rootstocks, that is the M series that has been selected by the people in Milan, but M4 is the most interesting one. And as you can see here, you have basically the same variety. This is Sangiovese grafted in pots on M4, SO4, and you would see that the very susceptible SO4 is really showing bad symptoms as compared to the other one. But if you want to look at numbers, this is what I would like to see more often when we impose the water stress on either well-watered and water-stressed SO4 grafted or M4 grafted. Well, you see that actually the canopy water use efficiency was really interestingly increased on the M4 grafted vine by 22%. And that is exactly what we need, because what we need is this. We need basically having a rootstock that either at a given water use would actually maintain higher photosynthesis, or vice versa, meaning that it would actually save water at the same photosynthesis. And this is very important in our environment for a very simple reason. Because in our environment, sooner or later, in summer, some rainfall is coming. And so in many cases, the issue is actually being able to overcome a couple of very severe drought, okay? A couple of weeks of very severe drought in summer, okay? And if you are able to, let's say, make the day, we can actually try to solve the problem without actually giving any water. By the way, our hillside land is not provided with water. I mean, the blue water is very scarce, and so it's very poor, and that's another issue. Well, this is just, maybe too detailed, but, when we compare the behavior of this SO4 M4 with the water stress and well water, of course, what do you expect to see from a rootstock that would give some tolerance? You would expect having, like in this case, higher breaks and maybe higher color and phenolics. That was the case. Whereas you see that the behavior of SO4 is truly a disaster, because there is no increase in sugar, although the yield was decreased, and no increase in polyphenols. So there is a difference. It's not a revolution, but it helps a lot. Then to get to the end, I know that we have a fairly strict time, but this sort of, trivial compound that is a kaolin is a very simple, this white clay that has this very nice feature of having a very low cost. And once it's sprayed on the canopy, it would actually affect light reflectance. There is a cooling effect. But again, we wanted to go into a little bit more deeper into the physiological response. And this is a fairly recent paper published. And basically what we saw, well, some were sort of expected responses. But these two were interesting. Here you have, for instance, upon a water stress that was established here and relieved there, whereas this is the time when kaolin was sprayed, this is the FV-FM ratio for chlorophyll fluorescence. And you see that basically at the end of stress, whereas the SO4 water stress were actually, the SO4 non-sprayed water stress really went down, and there were some leaf yellowing coming out at that time. Well, the kaolin sprayed were actually maintaining a non-limiting ratio. That is quite important, because that would be a sort of, life-saving mechanism. And as a matter of fact, this is a comparison between, you see, kaolin sprayed and non-kaolin sprayed on a water stress, okay? You see that there are two things important. Okay, this is the beginning of stress, this is the rewatering. You see that at the end of the stress, either way, the kaolin sprayed leaves would maintain higher photosynthesis. That is good. But it is very important that upon rehydration, you see that the kaolin actually goes up faster, and so the recovery is also faster. So again, if you are looking for something that is able to allow us to overcome the critical period, that might be, again, very helpful. Well, that's it, I guess. I think I have been using my 16 or 17 minutes. Well, thanks for your attention, and of course, I'm available for questions.
Thank you, Stefano. Maybe we can have one question Anyone would like to intervene?
I have a question, Aharon,
Go ahead.
if I may. Stefano, I'm curious, the increase in water use efficiency that you saw in the M4. What are your hypotheses to what do you attribute that to?
Well, it's a tricky question, Greg, because you perfectly know that if you look through the literature, there are quite, controversial explanations about that. However, my feeling is that actually the M4 is actually able to maintain the same level of photosynthesis with lower water use. This is what we see. About the mechanism, well, that really is still a challenge. In some cases, it can be root hydraulic resistance. In other cases, some people have hypothesized that, aquaporins are involved or other things. But, the thing I like about the rootstock is we have been trying the rootstock on different varieties, different environments, but basically that effect on the water use efficiency is very constant. It's very, that is really what we need, because we are totally harmless with rootstocks, totally harmless, absolutely. Another thing that I noticed, Greg, just to complete the answer, did you see the picture showing, the basal yellowing in the SO4 grafted and the green leaves in the basal green in M4? Well, that is another kind of effect. For instance, that variety was Sangiovese, okay? Typically, Sangiovese under stress would actually undergo a yellowing of the basal leaves. But apparently, that kind of process is inhibited when you graft on M4, okay? So it is actually able to change a lot the behavior of the of the vinifera variety. So again, it's not a revolution. It's not a revolution, but it's a promising tool. It's a promising tool.
Very nice, Stefano. Thank you very much.
Sure.
And we are going to move to Canada.
Wow.
Wow. With Simone Castellarin from the Wine Research Center, the University of British Columbia. Welcome, Simone. And Stefano, if you could mute your mic.
Yeah, sure.
Simone, are you there? Cannot hear you.
Okay, I'm here. Sorry about that.
Okay.
Getting ready with sharing the screen here. I had a bit of an issue. All right. Can you hear can you see my screen here?
Yes, we do.
Okay. Thank you very much, Aaron. Thank you very much for allowing me to present this work. So good morning, good afternoon to everybody. I'm connecting from Vancouver, the Pacific coast of Canada here. And today I'm going to talk about developing regulated deficit irrigation strategies for the Okanagan Valley. That is one of the major viticultural regions in Canada, and probably the driest. So our lab has been working for several years in understanding the production of secondary metabolites, particularly those metabolites that control the quality of the grape and wine. And we had a focus for several years on phenolics, anthocyanins, and tannins. But in the past 6 years, we started analyzing more carefully the aroma compounds, those compounds that are volatile and affect the aromas of grapes as well as wines. And most of these compounds, as well as phenolics, are produced in the skin of the grape. And so an interesting feature is that when we play with the size of the berry, possibly by regulating irrigation, then we can increase or decrease the concentration of these compounds that are produced on the surface of this barrier. However, we have also seen over the years that by applying deficit or applying regulated deficit irrigation, we can affect directly the biosynthesis of several compounds that are produced in the barrier, and including mostly phenolics like anthocyanins. As I said, in the past years, though, we focus on volatiles and aromatics, and particularly we focus in this group of compounds that are called terpenes, and they affect the aroma of very important varieties like Gewürztraminer, Muscats, Rieslings, etc. So we normally, or in many dry areas, we apply regulated deficit irrigation to tune the quality of red grape varieties. However, we have less information about what happens when we apply stress or moderate stress on white grape varieties, and particularly on the aroma components of those varieties. We started years ago in collaboration, a collaboration between University of Udine with Ben Gurion University, Dr. Fait and Dr. Mativi at the Mach Foundation. And we started investigating the effect of severe water deficit on this white grape variety grown in northeast of Italy, Toccai Friulano. And here you can see, well, how severe was the deficit we were applying on the figure on, below here. And we were comparing the fruit composition of these plants exposed to severe deficit to plants that were well irrigated. And one interesting feature was that we observed that during ripening time, there was an increase in the production of some terpenes, alpha-terpineol, nerol, under deficit irrigation. This increase in the production was supported by an overexpression of several genes that are involved in the biosynthesis of these compounds. So we thought that was interesting. However, this practice was difficult to sustain in a commercial vineyard. It was very severe stress, as well as we had obviously quite severe impacts on the yield of these plants, with decrease in yield, as well as you can see on the canopy. So interesting though, we had also produced wine from these grapes, and we observed that the same thing we were seeing in the grapes were translated into wine, with wines with higher concentration on terpenes, both in the free form, that are the ones that are volatiles, and the bound forms, those are the terpenes that are bound to sugars molecules, are not volatiles, but they can become volatile after, during fermentation or during aging, when this bound is cleaved and terpenes become volatiles and might affect the aroma of the wines. And we also observed that there was a higher intensity of some aromatic components of the wines. Again, as I said, though, the problem was with that trial for us was a severe deficit and probably not sustainable in commercial vineyards. And now we go to today's topic and today's focus, that is about the studies we have conducted in the past 5 years here in British Columbia, Canada. One of the major viticultural regions of Canada is the Okanagan Valley, a dry area that the Osoyoos Indian band calls Inkamip Desert. It's actually not a desert, it's a semi-arid shrubland, but definitely very dry, with limited precipitation during the growing season, from 100 to 130 millimeters of rain from April to October. And here we sustain production of grapes just by irrigated from blooming time to harvest, with quite frequently. And in this context, we do apply regulated deficit irrigation for growing our red grape varieties, but we normally have full irrigation, and we do not apply any type of stress to white grape variety. And this study was focusing on a variety we used to produce white wine, like Gewürztraminer, and we investigated the effect of regulated deficit irrigation, so that we were managing a moderate water deficit in our vineyards. and we focus on the impact of these strategies on the physiology of the canopy as well as the yield, but then with a strong focus on the composition of the fruit at harvest. Just as a bit of background on the growing area, this study was performed in Oliver, British Columbia, and here you see the growing degree days of the region. So it's 1600 Celsius at the end of the season, and you can quite consistently across the 3 years where we perform the experiment. So it's not really cool viticulture. Actually, this area is the warmest in Canada, and during the summer we can hit temperatures that are above 30 for sure, and sometimes above 40 degrees Celsius. Here the rainfall, and you see we have rains until June, but then we have dry periods, and we can we have to sustain the vines only with irrigation. The study was performed in Gewürztraminer in a vertical shoot position vineyard, and it has been recently published a couple of months ago, and so if you are interested in some of these details, you can find them in this publication. What we did was to apply several irrigation treatments. The control was the strategy that wineries normally apply to produce white grapes. So no water stress and frequent irrigation in order to sustain yield during the season. We applied 3 regulated deficit irrigation treatment, what we call early deficit, with plants being exposed to moderate water deficit before veraison and then well irrigated after that. Vice versa, late deficit was no stress before veraison and then moderate stress after veraison. Prolonged water deficit was consider non-irrigated plants during the whole season. Obviously, we as I say, we want to impose a moderate water deficit, so we withhold irrigation, not completely. And in fact, we use leaf water potential to monitor the plant water status, and we apply irrigation every time that the plant goes, or the leaf water potential goes below minus 1.3 megapascal. That was our threshold for leaf water potential. Versus the control, and the well irrigated vines were kept at water potential levels that were above or equal minus 0.8 megapascal. So what we did was considering the effects of these treatments of the physiology of the canopy and yield and sugar accumulation during the season, as well as the composition of the fruit at harvest, and particularly we focus on free and bound terpene profiles. Here is a graph that summarizes the irrigation amount that we gave during 3 seasons to our treatments, and here you have in black color the controls that always receive the highest amount of water, and as I summarize here, our treatment had, resulted in a reduction of irrigation of 30% for early deficit, from 30 to 40% depending on the season for late deficit, and 50% for the prolonged water deficit. I'm not focusing on these water potential graphs, but basically it's how we manage irrigation by measuring water potential on a weekly basis and tuning water in relation to our measurements. As I said, well irrigated vines that just maintain the water potential around minus 0.8 megapascal, and deficit irrigated vines that had water potential up to or down to minus 1.3 megapascal. So the idea was to apply moderate stress and to limit the effects of this stress on the canopy physiology. But as you can see here, the application of moderate water deficit affected for sure photosynthesis, and overall this was quite consistent across the 3 seasons with a reduction of about 50% of the photosynthesis in the canopy. This is like, let's go to harvest time, and this is just a picture that shows how the grapes look like if exposed to prolonged water deficit. Again, we also applied to all the treatments like leaf removal, and so all the clusters of our vines were, I will say, fully exposed to the sunlight. And here are some of the data on yield, where we observed that, yes, by applying regulated deficit irrigation, we do affect yield. This is an expected result, but we affected yield only with 2 out of 3 treatments. So with early deficit and prolonged deficit, here you can see that early deficit and prolonged deficit had resulted in 9 tons per hectare versus control, and late deficit were around 12 tons per hectare. This is about what the commercial vineyard where we applied our experiments normally obtain. In terms of berry composition, we had a decrease in the sugar concentration with prolonged water deficit, and an increase in tetratable acidity in the treatment. We know this result is not consistent among varieties. We have performed several deficit irrigation treatments with different varieties, and we observe sometimes increases, sometimes decreases. In some cases, we observe no effect of deficit irrigation on the sugar concentration. But interestingly, with Gewürztraminer in our region, we observe consistent results, and those were always pointing toward decrease in sugars and increase in acidity, titratable acidity, when we apply prolonged water deficit. So going to summarizing some of the results we had on the volatile components, so the aroma components of the grape. Here is, here I'm plotting the total terpenes as well as the concentration of 2 main terpenes that Gewürztraminer produces in its grapes. We did a profiling of about 20 free terpenes and 15 bound terpene. Here is the result that we, I took some of the most interesting results. One thing that is that the total amount of terpenes was not significantly affected by regulated deficit irrigation. However, we saw consistent results among 3 years on the toward an increase in the concentration of geraniol and citronellol, that are 2 main terpenes. Geraniol is the most abundant, but both terpenes contribute to the aroma of Gewürztraminer grapes. And so late deficit was the strategy that improved the concentration of those terpenes, and control was actually the treatment that had the lowest effect, the lowest concentration. However, early deficit and prolonged deficit had no effect on the concentration of these terpenes. And the same was observed for bound terpenes, the ones that are linked and attached to sugars. We observed no effect of our regulated deficit irrigation treatments on those terpenes. And this was consistent across the 3 years of study. So before I go to my conclusions, I hope I still have a minute. That was what I just showed was the results from our experiment on Gewürztraminer, while we performed another study in the same area on Viognier. Viognier is one variety that's getting popular in the new world viticulture, and we produce this white grape, this fairly aromatic and characterized, or the Viognier wine is characterized by a stone fruit aroma that is due to terpenes, actually. And in this study, we applied, again, regulated deficit irrigation in a similar manner that I just showed, for Gewürztraminer. In this case, we didn't have a late deficit treatment. We had only early and prolonged deficit. And we did observe that early deficit was increasing the concentration again of some terpenes like linalool and alpha terpineol that is quite important for the stone fruit aroma of this variety. This was consistent again also with changes in the expression of several of the genes that are controlling these the biosynthesis of these compounds. So as a conclusion of our study in the Okanagan Valley, we found that we can apply regulated deficit irrigation also on white grapes. This has, well, allows to save water. With our threshold, we saved 30 to 40% of irrigation water with no impact on yield, particularly when we applied deficit after vibration. So the late deficit for Gewürztraminer was the one that had no effect on the yield. We can affect the concentration of important compounds like terpenes in the grapes, particularly free terpenes. And but we observed that this is a response that is variety dependent. So the type of terpene that is affected is different among varieties, and makes sense because varieties have different terpene profiles as well. But also the timing of deficit irrigation might result in different results when we consider different varieties. So the conclusion of our study is that we can use regulated deficit irrigation also as a tool to manage the quality of white grape varieties in dry viticulture regions. So I'm done with my talk, but I would like really to thank all the people at the University of British Columbia and our industry collaborators and Andrew Peller Winery, as well as MRS Vineyard Services, as well as our funders like the BC Wine Grape Council, MITAX, and BC Investment Agriculture Foundation. But particularly, I would like to thank, with this, I conclude my presentation, Eugene Kovalenko, the master student that conducted most of the experiments I've been presenting today. So thank you very much, and I'm happy to answer any questions.
Thank you, Simone. Unfortunately, well, we can try to squeeze in one question. Anyone?
Very quick.
Stefano.
Yeah, very quick. Well, Simone, if I add those results, I would be very happy. Because if you are able by RDI after aeration to not affect yield, that is not surprising, maintain sugar and improve the aroma, that's really an ideal result. But I think that there is a picture that you showed of that row with this nice canopy and all the cluster exposed. Well, you know, that is a very efficient canopy. Because if you estimate the leaf area to yield ratio, that is a ratio that gives you a buffer also. I think that some of the reason why you had excellent results is because you're starting from a very balanced vineyard. And even if you impose some stress after aeration, you're actually getting the best of the stress effects and you are limiting the most negative ones. So it's also a matter that you have an excellent management of that vineyard.
Okay, that was a very good comment. No time for answers, Simone.
No, I can just agree with Dr. Corney.
Thumbs up. Okay.
Thumbs up, yeah.
Okay, now we move to France. I guess you're in France, right, Greg, currently?
Yes, I am.
Okay. And Gregory Gambetta is with us from the Institute this is work from home.
Live from my kids' room. So thanks a lot, Aharon, and thanks to all the organizers of the Triple D for inviting me to speak. I hope you can all see my presentation. So today I want to make a few points. First thing I want to do is introduce a framework for vine water use behavior under drought. This is the first thing. And then I want to use this to discuss a couple of points. And the first thing I want to talk about is the variability that we see between cultivars. and how this kind of intersects with the variability we see due to development or due to environment. I'm going to call that plasticity. So how does the difference between varieties that's innate kind of intersect with the plasticity that we see? And then finally, I want to just give an example from our lab about combining traits to see a complete picture of what we mean when we say drought tolerance. So, you know, Stefano already talked about drought. Simone talked about drought. We want to search for drought tolerant cultivars and our rootstocks. And when we do that, what do we mean? What is the target? So this is going to be the framework that I'm going to use today we're going to see a lot of data that kind of falls in the same framework. On the x-axis, we have the level of stress, you know, as expressed by a water potential getting more negative. And then I always think about the sort of stress response spectrum in two phases myself. One is this productivity phase, and this is where we spend most of the time in a vineyard. And then we get out here, if we keep going deeper into the stress, we get into some mortality risk. And as we kind of go along this spectrum from less to more stressed, we can identify some traits that may be interesting to us if we're thinking about something like drought tolerance. So if we look at water use, you know, you start off well watered, we have maybe maximum transpiration or maximum stomatal conductances. These may be different between cultivars. How we regulate water use with stomatal conductance can be interesting. Of course, probably most of you know, this is super widely studied. Eventually, stomata close, right? So this can be kind of represented by this other trait called the Turgor loss point, which kind of embodies the vine's ability to fight against the stress by accumulating solutes and maintaining open stomata. Maybe that shifts back and forth. Maybe some cultivars can tolerate more negative water potentials than others. And so these are all working kind of to keep the vine productive. When we pass this point, other stuff happens in the vine, right? We can lose leaves. Stefano was talking about this. This could be due just to accelerated senescence. It could be due to embolism though as well, which is when you get the water columns basically under so much tension that the water column breaks and you get little air bubbles forming in xylem. And this can drive leaf fall too. And then you can get this in stems. Now, grapevines are kind of peculiar in that they have this nifty little quality that's called vulnerability segmentation. And that means that the leaves are actually more vulnerable to forming embolisms than stems are. So grapevines are built in this way, so they'll drop leaves preferentially, and this protects stems from more negative water potentials. So we can think of, you know, any of these traits may be interesting. This HSM is called the hydraulic safety margin, and that's kind of like the distance that a given genotype would have between when it closes stomata and when it starts losing leaves. And I'll show you later that maybe this could vary too. So all this stuff can vary. But I think what we're interested in viticulture is this generic scenarios. I just made up these two scenarios as kind of a thought experiment here. That we are constantly talking about different cultivars and their behaviors. And so we can have a situation where different genotypes are really distinct. And this is kind of my low plasticity, the top example. So, you know, genotype A behaves one way, genotype B has a different quality. We could talk about drought tolerance here. And the plasticity that you get within a season or due to environmental acclimation or something isn't that great relative to the differences between cultivars. And that would be a situation where we would have to reach out and change a cultivar to get a completely different behavior. But there's an alternative way of looking at this, which is that maybe the plasticity is really high relative to differences between cultivars. And if that's the case, it changes a lot of stuff. It changes a lot of our decision making, because we have a situation where if we understand that plasticity and we can leverage it, we can maybe push the behavior of genotype A over close to some other genotype, and we may be able to save ourselves from changing cultivars. The idea is very hip in viticulture and climate change viticulture that we're going to always use more exotic cultivars. We're going to change cultivars. I would argue that that maybe isn't the most realistic solution, since markets are pretty constraining when it comes to cultivars and wine. So now I just want to talk about how variable is stuff, is a quality. And like, let's start, and we're going to create everything equal and say, how variable is stuff between genotypes? So I'm showing data here from one study by Alex Levin, who now works in Oregon, but he was at UC Davis getting his PhD when he did this work. And this just shows one trait, maximum stomatal conductance. It was done in a field-grown vines in a common garden. And we can see, if we can include Syrah, that there's about 30% variability in just the maximum stomatal conductance that these vines exhibit. Syrah is kind of an outlier, we can see in this study. And so if I limit that and I say, okay, let's ignore Syrah, we get about 18% variation. Now, it kind of depends if you're a pessimist or an optimist, I guess. If you think this is a lot of variation or a little, to me, I always think it's not that much. When I look at it, I'm like thinking, ah, God, you know, so to me, this looks like it's really not that much variation to me. So anyway, let's move on. So what are some other examples? So I just showed you this study by Alex Levin. In our lab, we have done a study was published this year by a postdoc, Sylvina Dyer, and then we will have another study coming with more unpublished data, looking at leaf vulnerability traits. And we get about a 35% variation in leaf vulnerability, or that would correspond to kind of the propensity to drop leaves under drought. So again, it's not huge. There's some variability there, but it's not huge. And then another study that we've done looking at stem vulnerability across, that one's across 9 genotypes. The stem vulnerability is across 22. So much broader range of diversity in the stem vulnerability study. We still only get 26% of variability. So you can see that the magnitude of movement that we get between varieties, all other things created equal, looks fairly limited. Maybe we would expect that because we're working within a single species. But anyway, it's food for thought. But if we look at plasticity now, switching gears to look at plasticity, there's a lot of studies that demonstrate, and these are more recent, that the plasticity is very large as well. This is a study from our lab that was done a couple of years ago, looking at the vulnerability of stems to embolism. And you can see this peculiar thing during the year, whereas in July, these are a bunch of different genotypes. This is the stems vulnerability, increasing embolism formation with decreasing water potential here. We use the P50, or the point at which you get 50% embolism, as kind of a benchmark value. You can see in July, it goes from about -2. Later in the season, it shifts, it gets closer to -3, and then you get a little bit more movement, barely any more movement, but a little bit in October that goes closer to -3. And so this is just, doesn't matter what variety, within a single variety, movement that you're getting across the season. It's about 25% variation. That's pretty significant. These are all just grown under the similar conditions. So this is pure developmental change. So a new study just came out from Uri Hochberg's lab that demonstrates a similar phenomena. And this is in other traits, and it looks exactly the same. So in this study, the group looked at the regulation of stomatal conductance, which is these solid bars, and leaf vulnerability, which is these dotted lines, and this point, I mentioned it at the outset, this turgor loss point, which are these little lines going up. And we can see that if they looked at May, July, and August through the season, you see these really big shifts. Here, the shifts in this study are really much larger than these 30–25% shifts. So the seasonal plasticity in this study is really, really big. So the take-home message here is that is there variability between cultivars? Yeah, of course. You know, when we look at traits, there is variability, and it's significant. I wouldn't say it's huge from what we've seen so far. That there's seasonal changes though, and I want to point out that they're always moving in the same direction. They're moving that from the vines being less tolerant to more tolerant. And so they're going through the season kind of with the expected weather changes. You know, this is kind of handy if you're a vine. And many of the seasonal variations look to be really large, as large or larger than anything that we see when we compare genotypes. Now, I have a little parenthetical phrase here to say, be cautious. One thing is we don't always, there's a lot of varietal diversity that we don't explore. We probably haven't leveraged all of the diversity that's out there. So it may be that the variation between genotypes could be greater. We tend to always use, you know, commercial varieties. In the work of my postdoc, Sylvina Dyer, she's used a lot more exotic varieties. And I'll tell you, we don't get a lot more movement or a lot more diversity when we use these more exotic varieties. They tend to lie within the spectrum. Those results aren't published, so. However, drought tolerance is more than just the ability to tolerate negative water potentials, right? So I want to leave you just with this one example of combining traits. So again, Sylvina Dyer, he's been just doing a ton of awesome work. Characterized a bunch of different traits in different cultivars, and I'm just using these two as an example. These are put in pots. This was in potted grapevines. So if you look at Grenache and in Semaillon, this is the data that she got in her experiment, subjecting them to a dry down, and then also measuring leaf vulnerability too, and putting these two traits together to get the hydraulic safety margin, or the distance between them. You see that GS in Grenache, the stomata close much more early than they do in Semaillon. Grenache has a propensity to lose leaves at much higher water potentials than, or less negative water potentials than Semaillon does, and it has a more narrow hydraulic safety margin. So if we only consider, this is still looking at more than one trait, it's looking at many traits, but when we look at the picture this way, we would come to the conclusion that Semaillon can tolerate much more negative water potentials than Grenache can. That's what our data says. And now, what does that mean? Is Semaillon more drought tolerant or not? And so conventional wisdom would say, no way, Semaillon is not more drought tolerant than Grenache. Grenache is more drought tolerant, right? But here we see that it can't tolerate as negative water potentials. So what's going on? So in the same study, we used modeling approaches to simulate dry downs between cultivars under exact same conditions to look and combined other traits. I didn't show you these traits in the previous slide, but I'm showing you here. This is transpiration on the axis, and this is just a really simple simulated dry down using a model that was developed by Hervé Cochard and colleagues. But you can see that Semaillon actually crashes, this is transpiration, earlier than Grenache. Grenache maintains transpiration longer, and then the solid line, which ramps up, is actually the loss of conductivity in leaves. We can see that Semaillon actually loses leaves sooner than Grenache. And this is because Semaillon, in addition to being able to tolerate more negative water potentials, also has a much higher Emax under well-watered conditions. So it depletes soil water much faster. And so when you look at the modeling results, it looks exactly like what you would expect from conventional wisdom, that indeed Semaillon is less drought tolerant than Grenache. I use this example just to demonstrate that combining traits is really important, and traits that are functioning when plants are not stressed may be the most important traits. This is really, I think, an important point. We do all these experiments, we always apply a drought to study drought. But why do, maybe we don't need to apply a drought. Maybe it's the traits that the vines exhibit when they are well watered that become the most important traits for their performance under drought. We can just screen vines without a drought. we can screen them under well-watered conditions and learn a lot about how they will perform under drought. So that is the conclusion. So my take-home message, phenotyping drought tolerance is, I'm still working on trying to figure out how to do it myself. So I think it's about integrating many traits and trying to get a complete understanding of how the thing's going to behave before we come to a conclusion about whether or not this genotype or that one is going to be more drought tolerant or less drought tolerant. And this is one of the questions that's kind of been haunting me most, is that, you know, a lot of these traits appear completely interconnected and they have a big amount of plasticity. So we need to learn about the plasticity, but we also need to learn about how interconnected they are and if we can even disentangle them from one another. Because if we can't disentangle them from one another, it really changes the landscape of how we need to approach these questions of what makes something drought tolerant and how we would potentially breed something for drought tolerance. So with that, I'm done. Thanks again for the organizers for inviting me. I wish I was in Israel and not in my kid's bedroom in Bordeaux.
Thank you very much, Greg, for this really interesting talk. So one very fast question, if anyone. If not, we move on to Ishai. Ishai Netzer is with us.
Yes, I am here.
Fantastic. Thank you, Greg, again.
Thank you.
So Ishai Netzer from Ariel University, and welcome, and I leave you the stage.
Okay. Good evening, good afternoon, everybody. Okay. So I'm going to present today the effect of late shoot pruning, also referred as post-bud break pruning on Malbec vines and wines. I'm from Ariel University, and my collaborators are from Carmel Wineries and from Hebrew University and Ben-Gurion University. It's not moving. Let's give it another push. I'll try to share it again. Okay. Okay. And my colleagues are from other universities. Didier Sued and Matania Ariel are the master students. Daniel and Sarel actually helped with the field measurements. Elishiv made the wine. Jose Greensweit from the Hebrew University is another collaborator with the non-structural carbohydrates. Aaron Fait from Ben-Gurion, secondary metabolites. Noah Hannah Levy on the statistics, and our collaborators from the industry, Gil Nir and Gil Harari. So these two students actually held this experiment for 4 years, 2 of them each, and again, my system is stuck, and I'll try it again, sorry. Okay, the motivation to this work was like this. Sometimes in hot places, arid places, especially with heavy yields, we are waiting for the bricks to reach values of 24, 25, while the pH is running away. So we can still wait, and the pH will reach 3.6, 3.9, and even 4.1, while we want it to be, the winemakers want it to be on about 3.5 and 3.6. So we thought maybe we will try to postpone the timing of the bud break, and then the maturation, meaning stage 3 towards harvest, will be also delayed, not to be on August, which is pretty hot in Israel, but to be on September, which is a little bit cooler. So actually this guy, Gil Harari, is playing with this idea for 10 years and he have a couple of data. And now we put everything in one organized experiment. We actually practiced 3 late shoot pruning or post bud break pruning. One of them applied 1 week after bud break, second 2 weeks after bud break, and the third 3 weeks after bud break. And we had 2 control treatments. One is winter pruning that we pruned all of them in the same way in the winter, but on February. And the other one is what we call control 2, is winter pruning, but afterwards we diluted the number of the clusters in a way that each shoot had only 1 cluster. So we're speaking about Vitis vinifera Malbec vineyard. The vineyard was planted during 2010. 6 years later, we started the experiment in 2016. It's a clay loam VSP. So we applied 5 treatments, 4 replicates with including border lines, and we gathered the data for 4 years. I will show only 1 or 2 years over here, but believe me, the data is consistent and the trends are rather similar over the years. So our vineyard is located down here between Tel Aviv and Jerusalem in a very ancient place called Ayalon Valley, more than 200 hectares of vineyard, mainly for quantity wine, not really high quality area. It's only 200, 300 meters above sea level. And here you can see our plot. You can see very, very easily the differences in the leaf area index and the canopy dimensions between the treatments. And if I apply the treatments, you can see all the treatments. Each replicate is consisted of 3 rows, and we are measuring only the 12 vines within the middle row. So we looked after dormancy and fertility and phenology, weekly leaf area index and stem water potential twice a month. Each season we performed 3 full physiology days, including stomatal conductance, stem water potential, etc. Yield and its components. Each of the 220 vines were harvested separately. We made each year 20 different wines, except for the first year, which we made only 5 wines. Our fight was done secondary metabolites, and Jose was doing the non-structural carbohydrates, and in my lab, we did the xylem anatomy. I want you to see this picture that was taken 3 weeks after bud break. We can see the winter pruning to the left in the bottom. We can see around LAI of 0.5, pretty reasonable. We can see the treatment that was pruned at the same day, the LSP3 in the bottom to the right. We can see much higher values of leaf area index, but in this day, these vines will be pruned. We can see to the left on top, we can see the LSP1 14 days after pruning. So there is start of the shoots, 3, 4, 5 centimeters of the shoots, and the LSP to the right on top, 7 days after pruning, we can see it's only starting the bud break. This is the cluster thinning made in May. Each shoot will remain only 1 cluster, and if the need, non-fertile shoot was also removed. Now to the results. According to Acorn and Lawrence, we reached the phenological stage. We can see the blue line and the black line of the thinning, the winter pruning plus thinning and the winter control, more or less the same. We can see the LSP3, which is the last to be performed. We can see how the phenology is moving toward the end of March. And then we apply the bud break and the phenology restart again to move on. The surprising thing was that during June, July, toward the horizon, all the treatments acted the same, meaning our aim to postpone the maturation or to delay the harvest from August to September, we failed in this mission. We are measuring a leaf area index with the delta T SunScan canopy analysis system. We can see a pretty the same picture. Let's see the LSP3, the gray line, raising up toward March. We applied, and then during April and May, the canopy still keeps rapidly going up until June, July, August, which more or less in all treatments, it's the same. Of course, in LSP2, which is the yellow line and the green line, it was earlier. Interesting thing is to see the difference between the control, which is the black line, and the thinning, the winter pruning with thinning, which have much more canopy. This is due to a phenomena that we saw all over the years of dehydration, a very vast, and you cannot, if you will fly with an airplane above the vineyard, you would see the control with a huge drought of the leaves, even though we didn't reach values of stress too much. We reached maximum minus 1.2, minus 1.4 megapascal, not more. And even in those conditions, which is not harsh stress, we saw a huge dehydration of the leaf, and I'll speak about it later. So in physiology days that we are measuring 5 times a day what's going on, you can see the first day, the second day, and July and August in the second and the third column. We can see assimilation rate and the LSP3, which the leaves are the younger, performed higher and net CO2 assimilation rate. And of course, the winter pruning with and without the yield were the lower. Same picture we see in the stomatal conductance. And something surprising is that the winter pruning, which was, of course, all over the season, was the most negative, it became the most less negative. That's because of the leaf dehydration. He moved the leaves, and of course, the stem water potential was improved. Of course, the photosynthesis was not improved. This is on the day of the first harvest. You can see the winter pruning, a lot of yield, a lot of very poor leaves, a lot of dehydration. We can see it to compare it to the winter pruning plus thinning. It looks much more nice. The canopy and the leaves looks much more vital. The LSP1 looks the same, vital and nice. The LSP2 is also okay. We can see a little bit of problem of a little bit late maturation, and the LSP3 needs maybe another week to be mature. I really don't have too much time, so I'll skip this, but in the TSS and the pH, we can see a much more united data that actually see that the connection between the TSS and the pH is much more favorable for the wine makers. We can see in the TA that as much as your canopy or is younger, you can have a much higher TA, which the winemakers actually wanted. If we move towards the harvest, and each vine was harvested separately, a number of clusters, we can see a very clear phenomena all over the years. Actually, we did this experiment also with Syrah, so it's the same picture in the Syrah. Of course, the yield was higher in the winter pruning and less in the winter pruning plus thinning. But as much as you perform the LSP, the late shoot pruning later, you get less and less yield, mainly because of the number of the clusters is reducing. Please pay attention to those two treatments, the LSP1 and the thinning, which have more or less similar yields, because I will want you to see the quality of the wine later. Second year, exactly the same phenomena. The only thing that was different is that the winter pruning itself had a little bit lower values because of the dehydration than of the leaves that we mentioned. If you look on the wine parameters, and I will not show you everything, just go to the total phenolics, we can see a very low quality wine in the winter pruning, improved in the winter pruning plus thinning, but a superior and much more better quality of concentration of phenolics that indicates the quality of the wine in the late shoot pruning. And again, it was as better as you move towards the LSP 1, 2, and 3. Dr. Shivi Drori is making the wine and making the organoleptic tasting. And you can see, and I will not show you all the parameters because we have a lot of parameters, but we'll see only the total score. This is the first year. You can see the score of 80. It's like $6 a bottle. It's very low. And you can see that all the LSP raised up to 4 times the value of the wine. In the second year, we had a replicate, so we can make statistics. We can see 81 and compare to 84 to 86. Again, more than about double the price of the wine in the same vineyard, same irrigation, same practice, just different pruning. So this one and this one, all the red circles are having the same yields, but improved quality, meaning the late shoot pruning is not only performing better because they have less yields, but also because of better photosynthesis, better performance of the vine itself. The very interesting thing is the dehydration. Everybody could see it. We count the number of dehydrated leaf. We can see in all the treatments, it's about 5–6%. And in the winter pruning, it is 42.3. And again, it was not too much stress, not more than -1.4 megapascal. So this kid is a high school teacher, and he made his biology project with us. He tended to be my son, but it's only by occasion. And he actually measured each vine, all the trunks, the diameter, and he picked the average diameter of the cane, not the trunk, of each vine. So he took them to the lab, and he cut them, and he stained them, and looked in the microscope, and analyzed the pictures with the imagery. And from the framework was that he can calculate the xylem area and the pith area of the vessel of the canes, can calculate the vessel diameter, the vessel density, and calculate the specific hydraulic conductivity and the hydraulic conductivity per one cane. And if you multiply it by the number of canes per vine, you can get a hydraulic conductivity in the level of the canes. Of course, it's all hypothetic measurements. So if we will look on this, on the specific hydraulic conductivity, we see that in the winter pruning, and this is, I didn't have time to put the statistics, but this is significantly lower in the winter pruning compared to the winter pruning plus thinning. If we would calculate it per cane, we can see that the winter pruning hydraulic conductivity is much, much less than the winter pruning plus thinning, meaning that the competition between vegetation and reproduction, the fact that in the winter pruning we had much higher yields, made a competition that led the anatomic system to be built in a way that I explain it, that we explain it, that the system could not move the right amount of water when needed, even though the situation of the stress was not stressed enough. So the hydraulic system was actually much smaller than it should be because of this competition. And if we calculate it for the hydraulic conductivity per vine, you can see it's sometimes up to 5 times higher. So this is one picture is better than many figures. You can see the winter pruning like this, and of course this is the average size, and this is the winter pruning plus thinning, much more ability to move water, and all the LSP is somewhere in the middle. So if I have to conclude everything, LSP, as much as you perform it later, you get also a foot thinning and improved photosynthesis. It costs less because you have less labor, and also we practice it this year. you have less pesticide because all the time of the powdery mildew, you actually shift and you escape the time of the powdery mildew and all the rains in the beginning of the season. The wine quality was undoubtedly increased. The delay of maturation in LSP 1 or 2, I can say that not. Maybe there is a slight delay in LSP 3 of maximum 1 week. And of course, in a way, we built the vine that will be resistant or not as problematic to problems of leaf dehydration. I have to make one comment. This is not good for all cultivars. I made it with Cabernet Sauvignon, 3 weeks. I got 2 clusters per vine. So each one who wants to take those ideas to other cultivars, do it very, very with precautions. I want to thank the Carmel Winery team, Oren and Dror, the farmers, Avner and Meir, all the technicians, Tom, Guy, Alon, Shachak, Yael, Roni, Yossi, Tamar, and Shiloh, and the people in our Institute of the Research Winery, Dr. Maria Staniewski and Nir Chen, and of course, for everybody that worked very hard in this project. And cheers.
Thank you, Ishai. That was very interesting. That's the best one. Best slide. All right. Okay, we don't have really time for questions. I'm sorry for that, but so we must move on. Maybe during the pause before the second session, we will have some time. If you want to stay, we can keep the stage open for discussion. Okay, now let me introduce you Kellen Gashu. Kellen Gashu is a PhD student in my laboratory. He collaborates under my supervision and Nurita Gam supervision. And we had a joint project with the R&D of Ramat Negev with Amnon Bustan. So he will talk about the variability among grapevine varieties in phenology in a desert environment in 2 different locations. Kellen, are you there?
Yeah.
Fantastic. So the stage is yours.
Allow me to share the screen. I don't know, so for some reason I cannot share the...
Maybe Shai should... okay.
I stopped.
Try again.
Yeah, okay.
Great. Open it up. Yeah.
Can you see?
Yes, perfectly.
Okay. Good afternoon, good evening, everyone, wherever you are. Today, I will present 3 years of data from the lab and field experiment and discuss how the temperature shift between the vineyards affect the vine phenology and its berry metabolism. I will start my talk by showing the evolution of global wine production and the areas covered by vines. Looking at this graph, the viticulture area is decreasing from time to time and consequently reducing the wine production. And the trend shows that the wine production continues to decline. This is partly due to the gradual shift of wine production from, the grape production from traditional area to newly suitable regions due to climate change. There is a nice paper about it recently published. They found that the 2-degree rise in air temperature can cause up to 56% loss of viticulture area within the current wine growing regions. Of course, here we can see considerable variability between the varieties in sensitivity to temperature region. In spite of that, the warmer regions are predicted to experience the greatest decline in viticulture area. As a result, efforts have been put to identify the most suitable climate zone for each cultivar. However, the genetic diversity among Vitis vinifera, along with the differential response to temperature region, prompts further investigation on the phenology and the berry metabolism. So the overall goal of our project was to decipher the interaction of phenology and fruit metabolism with environmental factors. To achieve that, we set up experiment in 2 vineyards in Ramat Negev and Ramon vineyard. In both vineyards, we had 30 cultivars, of which 10 are white and 20 are red. All cultivars were grafted on the same rootstock. We used the same trellising system. The soil was more or less the same for both vineyards. So we have tried to keep conditions the same for both vineyards in order to minimize the variation that comes from this factor. However, the 2 vineyards differed in their climatic condition. Ramat Negev vineyard experienced slightly higher temperature, both maximum and minimum, and here we can see a consistent 1.5 degree difference in average temperature between the vineyards was preserved throughout the season. We also computed the Huglin index, which categorized Ramat Negev vineyard as a hot and Misperamon vineyard as a warm region. Here, I want to emphasize that the spring in 2018 season was warmer than 17 and 19, which resulted the shift of all phenological courses to earlier in the season. For example, we can see this hierarchical clustering. The 2018 sample were separated from 17 and 19 samples. Oops. Not sure why it's not moving. I'm not sure. I don't know, it's not moving.
I guess there is a problem with the animation, so.
No. Okay, so here back to this slide. The 2018 samples separated from 17 and 19 samples. This interseasonal difference were mainly attributed to an earlier bud break in 2018 due to exceptionally high spring temperature, as I said before. What we see here is a summarized result of phenology. As you can see in this red arrow, the bud break, fruit set, rhizome, and harvest occurred earlier in a warmer Ramat Negev than cooler Misperamon. You can see this is the black line is Ramat Negev and the light gray is Misperamon. However, the duration between each phase are more important than the actual date of each event, because the climate factor during this period have a great influence on vine physiology and its berry metabolism. So here we can see the vegetative phase was longer at cooler Misperamon, whereas the fruit development and ripening phase was significantly longer at warmer Ramat Negev. And this result is true for both white and red cultivars. Despite this common response to temperature, there was also a significant difference between white and red cultivars. The white cultivars harvested earlier than the reds. They had also short ripening phases. So this prolonged pre-varizon and post-varizon at warmer Ramat Negev resulted berry shriveling in some red cultivars. For example, cultivars like Tempranillo, Ruby Cabernet, Pinot Noir, Dolcetto, the clusters in this cultivar just collapsed before harvest. Particularly, this cultivar, Tempranillo, never reached to the required harvest Brix level. In addition, the prolonged pre-verizon and post-verizon interval at a warmer region can increase rose accumulation. It can cause metabolic disorder or some other anthocyanin degradation, which will affect the fruit commercial value. In this respect, the white cultivars seems to be more resilient than the reds, because one, they reach harvest earlier than the reds. In addition to that, they have a very short ripening phase, which helps them to avoid most of the summer heat. However, having a short ripening period by itself do not guarantee a high fruit quality or wine quality. So it's important to see how this small difference in temperature affect the fruit metabolism of the white cultivar. So from now on, I will show you the results of white cultivar only. We measure the total carotenoid content at verizon and harvest. And here we show the total carotenoid degradation measured as the difference between verizon and harvest. And we can see from this bar graph, the degradation at warmer Ramat Negev was higher than at the cooler Mr. Ramon. Of course, there was a considerable variation between the cultivars and between the seasons. For example, in 2018 season, only Pinot Gris shows significant difference between the location. We also calculated the accumulation, the accumulated radiation from verizon to harvest for each cultivar, and we did a correlation against the total carotenoid degradation, and we found that an increase in radiation exposure was linked to a greater carotenoid degradation in many cultivars. Which didn't show this kind of correlation. Oops, I don't know. Again, something went wrong. I don't know. Sorry, something went wrong again. Okay, we also did a metabolite profiling on skin sample, and here what we see is that the variability between the cultivars at each location in their skin phenylpropanoid metabolism. And this PCA shows that the cultivars responded differently to the temperature region, particularly at verizon. If you see the first two PCS, at warmer Ramat Negev, the separation between the cultivars were more clearer than a cooler Misparama. Here you see cultivars were hardly separated. At harvest also, the separation between the cultivars were more evident in warmer Ramat Negev than Ramon. Despite this difference, there were also a common change between the vineyards. For example, this is a Pinot Gris here and Pinot Gris here, Colombard here, Colombard here. They separated from the rest of the cultivars. Good. And here we see, in the previous slide, we have seen the variability between the cultivars at each site. What we see here is the difference between the location in skin metabolites expressed as a fold change, Mr. Ramon against Ramat Negev. If you don't see this color key, the blue color represents metabolites highly accumulated at warmer Ramat Negev, whereas the red color shows metabolites accumulated at cooler Mr. Ramon. So looking at this heat map, we can see that the reduction of metabolites at warmer Ramat Negev were already observed at verizon. the reduction was even more clear and consistent at harvest. Of course, there were some exceptions among cultivars and metabolites. But overall, warm Ramat Negev was associated with lower skin metabolites than cooler Mispheromone. We have also tried to identify cultivars sensitive to the location and cultivars that are sensitive to both location and season. And here, the first piece here, we can see that cultivars like Semyon was sensitive to the location. Cultivars like Muscat of Alexandria was sensitive to both location and season. We know that fruits have a limited ability to open stomata and regulate their surface temperature. As a result, they face oxidative damage due to rust accumulation. And we hypothesize that even a small increase in temperature may impose higher oxidative stress in the fruit. And for that reason, we measure the H2O2 content at harvest, and we found that the H2O2 content at warmer Ramat Negev was higher than cooler Mispheromone. Of course, in every parameter that we measure, the variability between the cultivars were significant. So this higher H2O2 content also, higher H2O2 content can lead to degradation of other important compounds. So to explore this, we applied a correlation-based network analysis, and we found that an increase in H2O2 content in almost in all cultivars were associated with lower content of flavonols. You see this blue color represents a negative correlation. We can see also some amino acids in Pinot Gris, Riesling, Muscat Blanc, and Sauvignon Blanc were negatively correlated with H2O2 content, which suggested that higher H2O2 content in a warmer region can degrade or could lead to degradation of important volatile compounds. So to summarize what we have done so far, we have seen that even a small difference in temperature have a great influence on berry wine grapevine performance and berry metabolism. We have seen that the onset of the phenological event occurred earlier in warmer Ramat Negev. However, the fruit ripening phase were significantly delayed at warmer region. And slower fruit ripening at warmer Ramat Negev led to carotenoid degradation, phenylpropanoid degradation, and other resulted in, for example, high oxidative stress, which affects the content of, which related to the content of, which negatively correlated with the content of other phenylpropanoids. So in general, our results suggested that the biodiversity of grapevine will be a key in the reshaping of viticulture in warm climates. So the results of red cultivar are being processed. Even the whites, we have a lot of results. We didn't show all the results here. And having that said, we can suggest that Mispheromone desert could be potentially successful climate for wine production. Of course, not for all the cultivars. And this research was conducted in the supervision of Aron and Nurit in collaboration with Amnon Bustan. And finally, I would like to thank all the people who involved in my project. The sampling was a challenge, and all these people helped me in the field and in the lab. Without them, I could have not accomplished all this work. So with that, I finish my presentation. If you have a question, or welcome.
Thank you, Kellen. So this goes a little bit with what Greg showed before. the small variance in respect to drought sensitivity or drought tolerance or hydraulic behavior. Here we show actually that the cultivar very much affect or is very much affected by even a tiny increase in temperature, like can be a 2 degrees increase, while all the rest of the parameters are reasonably under control. So Greg, I must ask a question to you now. What do you think? How do you think we could explain these differences between different stresses affecting differently the variance between grapevine varieties?
I was getting to my unmute button. I don't know exactly, but I think that temperature drought interaction has to be one of the most pressing needs to be studied. We typically study drought in isolation and don't consider its effects with temperature, which is kind of naive. So I think maybe in the second session, there may be a talk on temperature effects, but yeah, it's something, at least in our lab, that we're just beginning to try to tackle.
All right. Okay, so we move on with the next speaker, Alain Deloire from Montpellier University. Alain, the stage is yours, and thank you for your patience. We're a little bit late, but it's all okay. Kerem, you should move out of the stage.
Yeah, normally from my side it's a video, so
yes,
I'm waiting for the video.
Yeah, exactly.
Stop sharing. Oh, okay. I'm out, no?
Yes, you are. Now I'm asking the technical stuff. Yeah, there you go. There should be some sound, right?
Yeah, we don't see. I don't hear anything.
No. No.
They should share the sound as well, not only the presentation.
Right.
So while you're sharing, you need to click share voice.
Climatic variabilities between years, a nightmare for vineyard management. Hello everyone. It's a great pleasure for me to share with you some information on the potential effect of abiotic factors on grapevine and berry physiology and development. This talk has been prepared with my colleagues, Dr. Suzy Rogers from Australia and Anne Pellegrino from France.
Wait, something is not.
So during this talk, I'm going to address some questions. For example, are climatic variabilities between years a nightmare for vineyard management and wine production? I suppose the answer is already yes. I'm going to speak about the effect of abiotic factors, mainly about water, but as well light and temperature on vine physiology and berry growth and composition. And then at the end, we will share some take-home messages. Speaking about wine business, wine production, as you see with this figure, it's about volume and obviously it's about price as well. So speaking about volume, you need water. You need water in the vineyard to manage the yield. Speaking about what we call the terroir, which is the interaction between soil and climate, as you know, water is a very important parameter, but not only. Temperature, light, wind, air humidity are as well crucial. And did you know that to produce, from a vineyard point of view, a bottle of wine, 250 to 350 liters of waters are needed? This figure is showing the annual rainfall variability between vintages from 1980 to 2008 in the south of France. And you see that the annual variability of rainfall is around 300 to more than 1,000 millimeters a year. So I guess the climatic variability between years in terms of heat waves, in terms of rainfalls, certainly are one of the most important aspects of climate change. And this is really impacting on vineyard management and wine styles, and on a yearly basis. But it's not only about water, it's about light, sunlight, and it's about temperatures as well. And this slide is showing many years of research on the potential effect of light and temperature on berry growth and composition. I really don't have time to develop, but you will have access to the presentation and you might want to have a look at those results. To summarize, I will say that for some compounds, light and temperature will stimulate their accumulation. For other compounds, light and temperature will decrease or will have no effect on their biosynthesis or accumulation. So, berry development and ripening, it's not only about the potential variability between vintages, it's as well a day and night story. In fact, researchers did show that some genes involved in berry composition are mainly and only expressed during the night. A few words now on the effect of vine water status on yield components. The rule of 70-30 will be presented. Grapevine yield components, it's a 2 years story. And the climatic conditions and vine physiology will impact year 1 and year 2 on yield components. So, from year 1, in terms of differentiation of primordia of inflorescences in the latent buds, up to year 2, up to berry set year 2, these are the 70% of the yield component. From berry set to harvest, the 30% represent the berry number per clusters and the volume of the berries. Let's see now how vine water status will affect vegetative growth and how this is helping to shape the vine architecture, obviously according to the training system which is used. The width of the canopy is due to the growth of the laterals, the secondary shoots, and the growth of the secondary shoots is under the control of vine water and nitrogen status. This study from Le Bon et al., published in 2006, clearly did show that the rate of leaf appearance of the primary shoots is under the control of vine water status in relationship with soil water availability. So the rate of leaf appearance of primary shoots is affected, starts to be affected when the soil water availability is around 35%. If you remember, I told you that the width of the canopy is due to the growth of the secondary shoots. And this study from Le Bon et al. Did show that the secondary shoots, laterals, are more sensitive to vine water status than the primary shoots. So the rate of leaf emergence in and for the secondary shoots starts to be affected when soil water availability is around 55%. This figure is showing the vine water status thresholds measured using the pre-dawn leaf water potential. And the thresholds are going from no water constraint to moderate water constraints and water stress. While moderate water constraints could be positive for vine functioning, reducing the vigor of the canopy, water stress has to be avoided at all costs, because then it will impact on the sustainability of the vineyard. So what to do in terms of vineyard management for wine regions or vineyards for which there is little water, available water? Knowing that 98% of the water, the needed water for a vine is transpired, the first step will be obviously to reduce the leaf area per vine. And because the leaf area per vine is reduced, the yield per vine has to be reduced as well to respect the ratio leaf to yield. Another tool we do have in the toolbox is obviously as well to reduce the plantation density, the number of vines per hectare, and to use small training systems instead of big training systems. This can be done up to the point from where the vineyard sustainability and the winery profitability are put at risk, obviously. The last part of this presentation will be about berry and water. Berry development occurs over two main stages: the green growth stages after flowering and fecundation, and the ripening stages, which is starting at veraison. The onset of veraison is berry softening. The volume of the fruits is under the control of vine water and nitrogen status, berry seed number, hormones, and other parameters. Trying to understand the effect of abiotic factors as water on berry composition, it's very important to differentiate pre and post veraison. Why? Because pre-veraison, during the green growth stages, some specific compounds are elaborated or accumulated, like tannin and organic acids. And to make a long story short, from veraison onwards, other compounds are accumulated. So, to make a long story short, it's important to understand the effect of abiotic factors in terms of berry composition, considering pre and post-veraison stages. This figure is showing that with irrigation you can control the volume of the fruit pre and post-veraison, and it's a way to manage the yield and obviously fruit composition and wine styles. A berry is accumulating water, but as well is losing water, very little by transpiration and potentially by water backflow. From the onset of veraison, which is berry softening, a fruit is accumulating sugar in green here and water, And this figure is showing the accumulation of sugar and water on a per berry basis. It takes around 26 to 30 days from the onset of veraison for berry sugar accumulation to reach a plateau. Then from the plateau of berry sugar accumulation, ripening is continuing, and if in theory the volume of the fruit is stable, sugar concentration per berry is stable. In most vineyards and for most varieties, from the plateau of sugar accumulation, in fact, the volume of the berry is decreasing because we think as an hypothesis that the phloem is not functioning anymore. And because the quantity of sugar per berry is stable from the plateau, and because the volume of the fruit is decreasing, the increase in sugar concentration is mainly and only due to berry water loss. So from the plateau of berry sugar accumulation, there is no direct relation between the increase in sugar concentration expressed in Brix here and the evolution of aromas and the evolution of phenols. Berry water loss and berry shriveling can be as well explained by the programmed cell death in berries. These research have been developed by my colleagues from Adelaide University and the National Wine and Grape Industry Centre in Australia. Berry cell death occurs mainly after the plateau of sugar accumulation and at the pulp level, and this phenomena is exacerbated by higher temperatures and drought. So I guess this is something we have to keep in mind speaking about berry water loss and berry shriveling. Berry shriveling is the ultimate stage of berry water loss. This is currently a huge issue in a lot of vineyards worldwide. Berry shriveling is obviously affecting yield and wine styles. So it's time now to end this presentation. This slide is showing that water, as we already spoke about, is needed from bud break to harvest. But as well, water is needed pre-bud break and post-harvest. Pre-bud break, the soil water content needs to be refilled to prepare bud break, and post-harvest, it's important to have functional leaves to allow the carbohydrate reserves to continue to be refilled. To summarize, the relation between vineyard management, cultural practices, abiotic factors, and fruit composition, wine composition, and wine styles are complex. And then my recommendation will be whatever you do in the field or whatever you do in the winery, it's very important to assess the effect of abiotic factors, cultural practices, winemaking practices on wine styles. It's very important to do wine tasting or to do some sensory analysis. It's a great pleasure for me to introduce you my colleagues, Dr. Suzy Rogers and Dr. Anne Pellegrino, who did help me to prepare this presentation. Thank you very much for your attention and keep safe and well.
All right. Thank you, Alain. Thank you very much for this overview of plant-environment interaction and their effect on wine. was very interesting.
So thank you very much to all the speakers of this first part of the viticulture session. We can start, and I am very happy to start the second session of the viticulture within the Dryland Desert and Desertification Conference. Just a note, on Wednesday, I think at 4 p.m. Israel time, we will have another session on viticulture, which is going to be more dedicated to remote sensing of viticulture. So you are, of course, all very much invited. We will have now a series of several talks, all providing different aspects on the GrapeX project, which is a large project that is going on in California for the past several years. I won't give too many details. Bill is going to present that much better than I do. So I would like to invite Bill Kustas from the USDA ARS Hydrology and Remote Sensing Lab to give us the first presentation of this session.
Okay, thanks, Nurit. Can you see this, my screen?
Yep.
Okay. So thanks for the introduction, Nurit and Aaron. Thanks for organizing this session, the viticulture session. So what I'll be describing is a project that looks at developing some tools for estimating vineyard stress, and with the ultimate goal of reducing water use while maintaining sustainability of our vineyard systems. We have multiple participants in this project. The list you see down there of various universities and NASA, but also a real important partner in all of this is Ernest and Julio Gallo Winery, which helped to facilitate this whole project. There are many, as I said, many cooperators here. Here's a list of a number of cooperators, some of which are going to be giving talks today. And it's just to emphasize that this type of work really needs an interdisciplinary group. GrapeX stands for Grape Remote Sensing Atmospheric Profile and Evapotranspiration Experiment. And because it involves looking at vineyards from both a micro meteorological as well as physiological, phenology, and how we can interpret vine land atmosphere interactions with remote sensing is sort of a core element of this project from a water management standpoint. So world grape production, it's encompassing all the various continents. It continues to grow. In California alone, there's reaching nearly 400,000 hectares of grape production, and worldwide it's expanding. But meanwhile, we have issues. In particular, in California, there's these frequent and persistent droughts that have occurred, most recently in the 2012 to 2014-15 time range. And what's happening is that there's been a major drawdown in groundwater storage without any sort of recharge. Even for wet years, there's no capturing of the lost water. You can see, especially in the southern central valley here, you get maybe 20 to 25% recharge during wet years, but during the dry periods, you're getting losses of 50, 80%. And so this is unsustainable, and it's been recognized as an issue in California and elsewhere. in the Western US, and policies are changing on how to regulate the water in especially groundwater systems. And so there needs to be a technology development as well as aspects related to discussions today about genetic work and other work on varieties to improve water use in vineyards. Total cropland in California is well over 9 million hectares just in California, and in almonds in particular is another large water user that has similar issues. So the motivation of GrapeX is really to get at estimating water use, evapotranspiration. It's getting actual evapotranspiration is important in terms of managing our water resources. And the way we've gone about trying to do this is using satellite and other Earth observations to get at the field scale estimate of water use. And the technique we've been developing is called the Alexi-Disalexi approach with a data fusion aspect that I don't have time to go into, but it uses multiple satellites at different resolutions to go from large scale observations from geostationary and polar orbiting satellites at 3 to 10 kilometers down to the 10 to 30 or 30 to 100 meter range is where the field scale needs to be to really monitor water use. So GrapeX started in a field near Lodi, California, started at 2 Pinot Noir vineyards, which are east-west oriented, and you see the information about their sizes here. And the purpose was to monitor and detail aspects of the vineyard to try to understand how water use evolves in these vineyards and to see how we can utilize remote sensing to reliably determine ET and stress from these systems. So we set out to put out a detailed set of measurements. Primary are these flux towers. These were towers in the northern and southern sites. These were different ages in the Pinot Noir. So we had a more mature in the north and a less mature in the south. So we could see some differences due to the age of the vineyard. These profiles had sonic anemometers at 4 levels, measuring the profiles because we want to understand how the vineyard structure itself could be affecting lower atmospheric conditions and the transfer of energy, water, and heat from these systems. We also measured heat conduction into these systems, which is quite different compared to other agricultural crops because you have this wide row spacing and clumped like vegetation. We were sampling at multiple locations here. These orange dots show you measurements of leaf area index across these systems to see how we capture this with remote sensing retrievals compared to ground measurements. And we also had spectral measurements within the tower footprints here shown here on the right-hand side where we did some more detailed monitoring and a host of other measurements, including soil profile measurements, infrared temperature measurements from towers, sap flow measurements, below canopy measurements, which I'll show you, as well as radiation measurements, which came to modify some of our modeling techniques. So besides some of these continuous measurements, these episodic measurements like leaf area index, below canopy measurements, photosynthesis measurements as canopy transpiration, solar radiation, and we actually had UAV coverage during intensive observation periods. And these would be periods that would be run in concert with satellite overpasses, so we get a range of scale of observations from the ground to the satellite scale. And besides those types of measurements, we also had some very localized leaf and canopy, as I mentioned before, as well as infrared temperature measurements looking at the behavior of temperature with both above and within row temperatures. So it was quite interesting information to add to what we were already measuring above ground, as well as one year we had these micro-Browen ratio measurements to look at conditions in the interrow. You have this unique canopy of both interrow and vine row conditions affecting the energy and water vapor exchange. We would focus these intensive observation periods to different vine phenological conditions, usually around these time periods where there was, of course, different grape conditions as well as vine conditions. And there's also different situations on how much water you would want to allocate both, during the controlling the growth of the vines and in periods before veraison and then afterwards affecting the vine or grape quality are aspects to consider. So one of the aspects of these measurements was to do some refinements of the land surface scheme that's within the remote sensing based model. It's called a two-source energy balance model. And it's sort of an ideal model to use in the case here where you have both vines and interrow, in this case, cover crop conditions, where you look at the resistances and transport both from the vine system as well as the soil substrate system affecting the total energy exchange with the atmosphere. Vines are really have unique canopy structures, as we all are aware of, where the biomass can be concentrated in the upper portions of the canopy. This affects the wind and radiation exchange as well as other aspects of energy exchange within the vine row system. So we were trying to capture all these phenomenons in trying to improve the development of the land surface scheme. One of the aspects here is in the vine architecture and how it affected the radiation exchange and extinction through that canopy system. And we looked at various modeling approaches and developed a sort of a refined algorithm that treats the canopy in a way that mimics what goes on in vineyard systems and to improve the way we do that. Another aspect we looked into was how the biomass distribution as it changes over time. There's a phenocam observations here just show you different aspects of vine development. Well, the biomass moves from very little, obviously, when just after bud break to being highly centralized in the upper canopy to moving more towards the central part of the canopy and developing a different type of wind extinction that more mimics what actually is going on in the canopy, air canopy layer. This compared to below canopy measurements shows an improvement using this more modified wind extinction. This in turn affects how the resistances are computed, and that in turn affects how the heat flux is calculated for this surface. So these types of improvements are now being integrated in the land surface modeling framework. With the UAV data, and there'll be some discussions on Wednesday about this UAV technology, but interesting aspects that are coming out are like LiDAR type data that come from the point cloud information from the UAV imagery, creating these canopy volume estimates that are linked to improving leaf area estimates as well as yield estimates. These are the kind of techniques that are being developed. So how do these improvements, what do they result in? Well, in the top figure there, you see in the original two-source model formulation and then modified with these extinction coefficients and improved formulations of the radiation and wind speed estimates, we get an improvement in what goes on in terms of estimating the energy balance components, the net radiation, the sensible heat flux, the soil heat flux, and ultimately the latent heat flux. There's also been a better development of the leaf area retrieval methodology, using some of the standard techniques of remote sensing retrieval has caused difficulty under certain conditions and certain vine trellis designs, and these improvements with the new algorithms have made for a better correlation, capturing the range in leaf area that one sees in these vineyard systems. Other aspects of interest when you get to high resolution UAV, there's development of shadow detection algorithms that were developed to improve energy balance estimates. As you can see down below you, this shadow detection actually changes the distribution of sensible and ground heat flux and latent heat flux estimates. So incorporating shadow detection can improve estimates when you're using very high resolution information. And this comes important when you're trying to distinguish vine from interrow systems. There's also the issue of separating the total ET, the total from the soil substrate and vine system, to isolating the transpiration from the vine system itself. And this plays into really categorizing vine stresses, which is what you want to ultimately get, and vine water use, and using these UAV imagery can provide a mechanism to get a better estimate of that. And developing those algorithms ultimately with satellite data is one of the objectives, to sort of do that partitioning at the satellite resolution. This is just an example over multiple years at the Lodi site, where we compare the red dots are the ET from the tower measurements, and then estimates from the footprint, which is the darker line. And then the shadow is giving you the range of ET produced by the model. And this really emphasizes the need, or the real advantage with using remotely sensed estimates of ET, is you really capture the variability that exists in your field that you otherwise cannot get with point samples. And so this is one of the developments that we're utilizing within this system. Here, what I just want to emphasize is that using observations from satellite can provide some operational estimates, but getting at the details, even down to the vine resolution and at critical times when we might not have satellite observations, might be best captured by UAV systems. And so the idea is that by combining observations from the ground through UAV and ultimately satellite, we can better capture conditions in the field and when we need to intervene for factors affecting vine development and growth. So the original site here, the Sierra Loma site near Lodi, we expanded to create a climate gradient from Northern California, where you have the Sonoma Napa region, down through Lodi and Sacramento, down to the southern end where vines are grown, to really capture varying trellis design, soil condition, and climate condition, to see how well these modeling approaches and retrievals that we've been developing at Lodi can be transported to these other sites. Not only from remote sensing, but from our ground-based measurements, we're also trying to understand where we hit these points of optimal water use efficiency using tower flux data and comparing, gross primary production to evapotranspiration. And so understanding how these vary across climate gradients is another aspect of this work. Here's just an example of what expanding the range of sites and climate, and where we're comparing now tower measurements with similar sets of instruments at these 3 sites and how they compare with our modeling system. Here showing the fusion approach with the red lines, the the diamonds are actual Landsat imagery, so sort of interpolating between those with our fusion model, as well as the ET observations, both energy balance closed and actual ET from the eddy covariance system to give you a range of what the actual ET is. What's interesting here is we get this climate variation. We still capture in a reasonable way the ET over the whole growing season and the interannual variability. The black bars there are the irrigation schedules, and then the blue are the precipitation events that occur. And you can see very different irrigation going on, plans and schedules go on from the north there. Here through the Central Valley and down through the Southern Valley, you see the more frequent irrigation and response of the vineyards to that irrigation. We're also testing the UAV observations across these 3 vineyard sites. And again, with some of these modifications, we're improving the ET, daily ET estimates using UAV technology. The same sort of partitioning we're also testing with the satellite data. And what you see here is a seasonal trends of the ratio of transpiration to the total ET, sort of peaking again during the peak biomass growth of the vegetation. These lines are related to these phenocam observations here to the right. And the yellow being the leaf area retrieval. And you can see at times we aren't capturing what the ground observations are seeing. And this is one of the research topics we've been addressing with improving the leaf area retrievals I showed you earlier. And we're going to rerun these analyses with the new leaf area retrieval algorithms and see how that changes the partitioning estimates and the total ET results. The other aspect we're looking at, which is irrigation management, which is sort of one of the goals of the GrapeX project and will be discussed on Wednesday, is capturing both stress and overwatering conditions that might occur in a vineyard. This particular study was in a variable rate drip irrigation system in the Ripperdan region. So these blocks here are 30 by 30 meter that are gridded irrigation blocks that can be controlled to distribute irrigation differentially across this vineyard site. There was an imposed stress in the northern part that was captured by the thermal imagery and the data fusion technique. And it also captured perhaps some overwatering that was occurring in the other part of the vineyard. This type of approach seems to be much more beneficial than using the sort of crop coefficient-based approaches using vegetation index procedures or approaches that can't capture this stress condition and is picked up well by the thermal imagery. So there's some challenges now that we're looking towards. Ultimately, this type of information to get a hands of the grower needs to be available to the grower either through their smartphone or their tablet out in the field. And so one of the ways we're going to try to do this is working with the Open ET project, which is coming up in the next years to provide this type of ET information to growers through Google Earth Engine and other means for providing useful information to the growers to make their decisions. Upcoming on Wednesday, as mentioned earlier, is going to be a remote sensing session on viticulture that describes many of the aspects that I could only briefly touch upon today as part of the GrapeX project. And so I hope people attend that. And I thank you for your time and attendance. The project is continuing and we look for collaborators who are interested to join our project and work with us on improving the techniques and that we've been developing. And I thank you for your time.
Thank you, Bill, very much. Quite overwhelming. I think we'll have to move directly on to the next speaker due to time constraints. And hopefully we will have some time at the end to have some discussion. So our next speaker is Andrew Macaron from USDA ARS in UC Davis. And Andrew, the floor is yours.
Thank you, Nuri. Good day to everybody. Thanks for organizing this and bringing us all together in these strange times that we're all dealing with. Going to be a link between the current session, talking more about grapebacks and the previous session, and really focused on how the plant is regulating the water uptake and transport that ultimately ends up in the ET signature that Bill was talking about previously. So I'm going to be talking about work that we've been doing over 10 plus years for this. And this is all in the context of sustainable water use for viticulture. So we're ultimately trying to develop ways to maintain quality and yields with less water and keeping the crop economically viable. In wine grape production, things that we've heard about earlier today, the irrigation management often uses deficit irrigation. And we want to know how much water, like Bill was just talking about, but also the stress level. And that's really determined by what's going on inside the plant in order to improve quality, in order to improve uniformity for harvest and reduce hedging and other things like that. And so as a plant physiologist, I think about the system similar to what Bill just described as the two-source energy balance, but from how the plant is regulating that transport along the soil to plant to atmosphere continuum. So what are they doing and what is changing inside the plants that ultimately impacts the amount of water that goes from the rooting system through the vines and up to the canopy where it's exchanged for carbon. And so you can think about the plants similar to a hydraulic model, a series of hydraulic resistors here that's shown on the right. And those hydraulic resistors are actually changing dynamically. And so ultimately what we've been looking for is what are the weak links within this chain of hydraulic resistors throughout the plants and the work that we've been doing. So I'll be focused here today on work that we've done looking inside the plant. So starting in the xylem, and then more recently focused on work on fine roots and up into the leaves and determining what it is that's changing amongst these hydraulic resistors and the conductivity in them. So as you're all aware, the water transport within the plant is driven by the cohesion tension mechanism. And just as a quick review, what's happening is when the stomata open in order for carbon to come in, they're losing water via evaporation. And that evaporation actually creates a pool on the water. The water is cohesive and creates continuous columns all the way down through the plant to the soil of where it's pulled in. And that cohesive water can actually withstand tension. And because it's under tension, it's actually considered metastable and can break inside there. And it actually breaks, as Greg and others alluded to this morning, via embolism. So we have air seeding that happens. You have the water column that's under tension, and it pulls a small air seed into a water column under tension, and it breaks the cohesive properties of that water column. It snaps and air bubbles fill that up, and you reduce the conductivity of that tissue. So grapevines were actually thought to be very susceptible to drought-induced embolism. And this idea actually came from, in some ways, curves like this. This is a hydraulic vulnerability curve that was generated by researchers, Anna Jacobson and Brendan Pratt, where you're actually looking at how much loss in hydraulic conductivity you get within a stem segment relative to drops in water potential. So moving towards the left here is increasing drought stress on this curve. You can see that you're losing conductivity as you move along that drought stress. I'll show this figure for different organs throughout the talk. But what's really key here, and this goes back to some things that Greg talked about too, is a parameter that we think of a lot for these curves is the psi 50. So it's the water potential at which we lose half of the conductive capacity. So a significant amount of that conductive capacity. In this curve, you'll see here that the psi 50 was minus 0.4 megapascals, which is very, very vulnerable to cavitation. That's saying that by 4 megapascals in the field, which you almost never see in Mediterranean climates during the growing season, that they've lost 50% of their capacity. And they've lost the vast majority of it by the time that irrigation managers are starting to get nervous about severe stress in these situations. So around the same time, we actually started using advanced visualization techniques with magnetic resonance imaging and comparing that to techniques where we would actually cut the stems. So the last curve that I showed you on the previous page is actually done by excising stem segments from the plants themselves. And then you measure the flow through that tissue, the conductivity of that tissue at a low pressure head, and then you flush it with high pressure to get the air bubbles out of there. And it tells you how much air embolism is actually reducing that conductivity. So again, we have percent loss of conductivity here, or the number of empty vessels that we see in some of the visualization techniques. And the key here is that the intact plants measured with MRI, you can actually see that their curves end up looking very different. And the Psi 50 value for these curves, depending if you're simulating that embolism with an air injection sleeve, like shown up here in the right-hand corner, or actually spinning the stems with a centrifuge to generate tension and simulate the tension, or if you're actually measuring it intact, I end up looking much more resistant to embolism under these intact conditions. I'll come back to that idea. So around the same time, we also started doing a lot of X-ray micro-CT work at the Advanced Light Source down in Berkeley. And we started doing this on excised stems. And Craig Broderson will actually talk about this in a talk on Wednesday in a section on water flow in plants and soil. But ultimately, we can look at things with much higher resolution compared to the MRI. And we started doing this on excised stems and looking at the xylem network and doing this modeling. But we figured out how to stuff a living grapevine inside the chamber where we do these x-rays. And you can see that down in the bottom left. And we've modified this chamber quite a bit. And so the image on the right-hand side is a cross-section of this stem of a potted grapevine that was subjected to drought stress over time. The colored vessels in this stem segment represent embolism as it was forming relative to its water potential, shown here in the center of this. And you can see that most of the embolism started showing up around minus 1.8 megapascals, which in grapevines in the field would be very stressed conditions for a stem water potential. We actually didn't see in many of the plants incipient embolism show up until about minus 1.2. And so going back to the Psi 50 value again, the grapevines were looking much more resistant to cavitation than was previously thought from some of these other methods that were being used. We also then, one of the questions that came up is, are you actually looking at the correct tissue? So are the stems resistant, but the petioles and other things are not? And we heard a bit about this in Greg's work earlier. We worked with Uri, who was a postdoc at the time with Missy Holbrook, and he was using MRIs, and we paired that with our micro-CT imaging. And what we found here is that the petioles were, in fact, a bit more susceptible to cavitation. But again, relative to where those other curves were actually showing huge losses of conductivity by 0.4 megapascals, there was very little embolism inside those petioles until about 1.5 megapascals. So there is this segmentation, but there's still a whole lot of conductive capacity well before that. So the xylem in the petioles don't look like it's actually limiting the transport in the system. So where does this idea come from? And actually, what I think, if you go back into the literature, we have this notion that grapevines are actually undergoing refilling of embolism on a regular basis. And John Sperry did some real classical work where he showed that grapevines were refilling winter embolism induced by freezing, which is a different mechanism that I won't get into. But that we all know when you prune grapevines in the spring, you'll actually see them dripping when the soil is quite wet. And so the idea is that they're pushing water through there, but whether that was actually happening within the season is where the question was at. And we also know that grapevines are hyper susceptible to winter embolism because of the size of their vessels. And so we translated this concept into drought stress, but it actually wasn't translating directly. Missy Holbrook did some of the first MRI work in 2001 looking at this, work from her lab. But what she actually showed is that you didn't get refilling of these vessels until water was re-added, the water potential climbed dramatically, and then the lights had to be turned off. So this was not actually happening during regular transpirational conditions. And we actually contributed to these notions that were out there. In 2010, we actually described the mechanism using CAT scanning of exactly how the refilling was occurring. We definitely believe that this is real and it occurs, but it's only after significant amounts of time and hypersaturated soil conditions that you would never see in Mediterranean climates in the field. We also demonstrated differences among species in terms of their capacity to refill. But again, it was only after water potentials really climbed very close to zero. And so refilling, in my mind, to clear this up, of embolism is only when the soil is hypersaturated and the vine has recovered to near 0 megapascals. And again, under Mediterranean climates, where most of the folks that are calling in from, this does not occur during most of the growing season throughout the world. We then also, just to pound on this a bit more recently actually looked at the petioles. Because the petioles being a bit more susceptible, we wanted to see within a daily time period of whether they were actually refilling. And so we actually drought stress plants, we induced embolism in there, and we actually looked at the recovery. And in a recent dissertation work by Catano Albuquerque, we found that there was really no rehydration or embolism refilling in these mean values that are shown here. And this is counter to some of the ideas that were put out there by Hans Schultz. He had a paper in 2003 that's really a seminal paper. But in the discussion, he actually says that maybe it's actually just a perfect balance between cavitation occurring and embolism repair that's actually going on in the petioles of these plants. And actually, there was another paper in 2011 by Vivian Zouffari and Hervé Cochard, who's on this, that they actually showed that grapevines were reaching 90% loss of conductivity in their petioles at midday and recovering by the next morning. But since then, Hervé, two years after that publication, came out and published this one, where he said hydraulic failure and repair are just not happening on a routine basis in plants. And we have sampled over 20 species now that we've been looking for embolism repair, and grapevines have been the only species that we've actually been able to document it in. And part of the problem with the measurements that were used previously is that when you're cutting off these stems under tension, what Jim Wheeler showed in a very seminal paper was that if you cut it under tension, you were actually inducing embolism into that tissue, and you had to treat it very carefully. And that at midday, when there's more tension in that xylem, you would get more embolism. So you would have the appearance at midday that there's more embolism and a recovery in the morning just due to this artifact associated. So ultimately from this, it really eliminates the xylem as key in terms of changing the resistance in there. Around the same time, we actually shifted into working on living tissues in leaves and fine roots. And that's what I'm going to tell you in the last few slides that I have here. So again, for Caetano's dissertation work, he was looking at a couple of varieties here showing Chardonnay, where he has stomatal conductance decreasing as we increase the drought stress. This is a curve similar to what Greg showed earlier, precipitous drop in that well before where we start seeing embolism showing up. And the leaf hydraulic conductivity, so measuring the permeability of that tissue, which incorporates the xylem and outside the xylem, was actually dropping at a very similar rate to this. So it looks like other parts of the pathway are actually much more important. Again, we documented embolism, no embolism in the leaf veins, as people said, well, maybe it's just further and further out in the system that you should be looking at it, and really seeing no embolism that coincides with this large precipitous drop in leaf hydraulic conductivity. And again, we verified that with micro-CT. So really what's going on is it's outside the xylem. Once water actually moves out of the xylem to the points of evaporation at these mesophyll cells inside the leaves, it's this pathway outside the xylem that seems to be driving that. And we used modeling recently to demonstrate that it's really about changes in plasma membrane permeability and Casparian-like strips inside the grapevine leaves that is known to exist under drought stress conditions. So we also demonstrated this in several other species to verify that what's actually going on is that drops in leaf hydraulic conductivity are much more tightly tied to drops in the outside the xylem conductance, and that the xylem conductivity actually is well maintained as you get this increasing drought stress. And I won't go into many more details about that. So in the last few slides, I want to shift to where the water uptake actually initiates, is at the fine root material inside grapevines, where we've been doing work for a similar amount of time. And we've actually been looking at how water actually moves across that cortex in the fine roots before it actually ends up in the xylem for long distance transport. When Greg Gambetta was in the lab, we were actually looking at aquaporins and the permeability of these tissues as it moves over into there. Also looking at suberization in the Casparian band and then in the epidermis as well. And what Italo Cuneo actually found during his dissertation, using the CAT scans again, is that he found that the xylem again was staying intact. We are not seeing embolism inside these fine roots. So this is a cross section of a fine root. But what we were seeing is that the cortex was actually tearing apart under drought stress conditions. And so this was actually limiting the hydraulic conductivity. So this is fine root hydraulic conductivity shown on the right axis. And that's what the black line is dropping precipitously with increasing drought stress. And that actually was coinciding with an increase in these lacuna. Other people around the same time, Jonathan Lynch at Penn State, was showing that drought could actually induce this air arenchyma in corn plants in a very similar way. And we actually mapped it and found that this was occurring some distance back from the root tip itself, about 5 to 10 centimeters back from the tip, and that the tip was actually staying intact and still functional in this area. So what we actually then did is looked at two different root stocks to compare that were known as drought intolerant and tolerant. And in a recent paper, we actually found that 110R, while both of them were forming lacuna, 110R, which is considered the drought tolerant, was actually forming these fine root cortical lacuna much more rapidly compared to the drought intolerant. And it was a little bit counterintuitive for us. We used neutron radiography then to image the root systems. We subjected them to drought stress and then we rewatered them and looked at the uptake around the individual roots. And what we found is that there was reduced elongation with drought stress, but then in the drought tolerant rootstock, we actually had this regrowth of the roots, and it actually coincided with 110R actually being able to take up more water around the root tips. And so Narit, this is my final slide here, is that we think there's several factors actually contributing to this. Is in these drought resistant rootstocks, what's actually going on is that they're forming more lacuna in these fine roots. And that is limiting competition for carbon that's being redistributed via the phloem to the tip. And it limits water competition. So the tip is actually preferentially getting water. Aquaporins are actually upregulated in these drought tolerant ones, and there's less suberization. So the tissue actually remains permeable, and it allows these roots to continue growing under drought stress conditions. And so I'll stop there. There were a whole lot of people involved in this work, but it really points to the living tissues in the leaves and in the roots as being the ultimate hydraulic resistor within the grapevine system.
Thank you very much, Andrew. One quick question, if anyone has any quick question to Andrew. Okay, so we'll move on to Mimar Alsina from E&J Gallo. Mimar, you can share your screen and put your video on.
Boy, I'll try. I hope it works.
Good morning or good afternoon. My name is Maria Mar Alsina. I work for E&J Gallo Winery in California. And I'm going to talk about a case study of evaluating a remote sensing-based toolkit to monitor vine water use and water status for irrigation scheduling in California vineyards. As you know, grapevines are grown under regulated deficit irrigation, and correctly managing the vine water status during the season is key to achieve the target's yield and quality. But there's so many questions that we don't know how to answer when we manage irrigation, like exactly when do we start irrigation? We need to ensure the availability of water early in the season or later in the season when we want to cause a certain level of stress. How stressed are the vines and how much water do I need to apply? Usually growers answer these questions by taking point measurements in the vineyard. However, vineyards are highly variable. It's strange that you have a vineyard that has homogeneous conditions in the whole block, like you see here in this example on the right side of the slide. So we think that having remote sensing techniques that can inform at sub-block level would really help improving the target.
Okay. Sorry. Can you hear me?
Yes.
I think the video is not working, so I'm just gonna.
We can hear you. You need to share the video.
Well, that's what I did, but I'm gonna try again.
Targets of yield and quality that we can achieve with irrigation management and
now you should be seeing
as responsible as possible with the water use. In other words, maximize yield quality and water use sustainability. Also, growers can take measurements of water status, for instance, with a pressure bomb. However, there's a piece of information that is crucial to irrigation management and that we haven't had in the past, and that is the actual evapotranspiration of our vineyards, how much water are using our vineyards. And this is what the GrapeX project that Dr. Costas has been explaining a moment ago has been focused in. And now I cite literally the aim of the GrapeX project is to provide the grapevine growers with a multi-scale remote sensing toolkit for mapping crop water use, actual evapotranspiration, and vine water stress for improved irrigation and scheduling in vineyards. So this is basically what I was saying in the slide before, delivering a toolkit that can tell us at a sub-block level how much water and how stressed our vines. So in this talk, I'm going to explain how we tried at Gallo to use the scientific results of GrapeX and turn them into practical tools. So we first of all wanted to get all the information that we could remotely sensed that is relevant to irrigation and put it together into a tool that we could deliver to growers. So, of course, we want to deliver water use. That's the start. That's what we didn't have before, and now we do at 30-meter spatial resolution and high temperature resolution. Then we want to give information about vine growth and water status, and finally, soil moisture. So we try to get all this information and make an irrigation recommendation based on that, and then test that against the current practices. We designed an irrigation pilot to do these tests, and we did it in two phases. Phase one was 2019, phase two was 2020. In 2019 was basically a test. One, we wanted to familiarize the growers with the remote sensing products. We wanted them to trust the products, so we provided them with information every week, but we didn't ask them to manage based on our recommendation or our data. However, they could compare with their decisions and their opinions. Then we had the opportunity to start designing and putting together a delivery interface and establish the workflows of data retrieval, processing, and delivery to be able to manage irrigation on real time. In 2020, in phase two, it was when we started asking the growers to manage a few blocks following our irrigation recommendations, and then we compared, is when we wanted to evaluate the performance of this remote sensing-based irrigation versus the commercial. As I said, the first thing was, let's put together all the information in a way that we can deliver the growers along with their recommendation. So we created this irrigation dashboard, we call it. This is the first tab that the user sees. This has the spatial information of actual evapotranspiration from the GrapeX remote sensing toolkit. We have the stress, two stress indicators. One is the actual evapotranspiration divided by the reference evapotranspiration also from the GrapeX toolkit. The other one is the normalized difference water index from Sentinel-2. And finally, we also have a layer of the normalized difference vegetation index as a proxy for canopy growth or canopy vigor. Here, the grower can browse over the maps, can zoom into a block, and can go all the way to pixel level and click and know the value. So we deliver this every week as an aggregate of the week. So the grower can click on a certain place in a block and know how much water the vines have used at that particular place and see how different that value is from another point, maybe in the same block. Here we see another tab where we see the soil moisture, in this case, based on soil moisture sensors that are installed in the ground. So the grower can click in a position and check what is the soil moisture at that point. And finally, this is a table view where we provide an average by block of the few spatial components that I showed before. And also is the table where we were providing the irrigation recommendations. Here are the details on how we did that. So in the experimental, in the phase 1, we selected 6 blocks, 5 Cabernet Sauvignon blocks and one Petit Syrah block, all in Sonoma Valley in California. These are commercial blocks. The grapes were harvested and they went to commercial wines. Blocks range in age and surface, and they are representative. They range also in rootstocks, and they are representative of the blocks in that area. This year, as I said, we scheduled the irrigation and provided to the growers, but we didn't ask them to use that recommendation. This year would be a baseline. We wanted to know how these blocks were doing, and we wanted the grower to get used to the remote sensing toolkit recommendation. In the second year, phase 2, what we did is we kept the 6 blocks that we had used the first year as baseline, and those we sent recommendations every week, and we asked the grower to follow the recommendation. And then we added 3 more blocks as control blocks. The ones that are in white, we added another Petit Syrah block, block 9, and then we added 2 Cabernet Sauvignon blocks. And these blocks, we asked the grower to just manage according to their expertise and their knowledge. Then, since we know that vineyards are variable and it's difficult to compare, we also added 1 block that we could split in half, and at least it's the same variety, same soil, same rootstock, and we irrigated half of it based on the remote sensing toolkit and the other half based on the grower expertise. In order to validate or to monitor the blocks, from May to October during the season, we collected the irrigation records. We had pressure sensors installed in each irrigation set. And then we monitored vine performance in both the remote sensing based and the control blocks. We did periodic measurements of leaf and stem wear potential, and also periodic measurements of leaf area index. To take these measurements, we selected between 3 and 9 spots per block, and the objective was to revisit each selected spot every 3 weeks. Sometimes was not possible because these are commercial blocks. So if there was any field management that didn't allow us to go in, then we couldn't sample. Finally, at commercial harvest decided by the winemaker, we took yield components for vine. So we cut and weighed all the clusters in each of these spots where we had been sampling. We took some of these grapes and sent them for a grape quality panel, an internal panel. And then we also got the records of yield per block recorded when the grapes arrived to the winery. We'll start taking a look at the water use. How were the irrigation and evapotranspiration in the remote sensing treatment and the control blocks? On the right side, you have 2 two graphs. The one at the top corresponds to the comparison of the two Petit Syrah blocks. So there's no error bars. The one at the bottom corresponds to the comparison of the average of the Cabernet Sauvignon blocks irrigated according to remote sensing or irrigating according to control. The lines represent the weekly actual evapotranspiration, and the bars represent the irrigation. The only thing I'd like you to see here is that there are not big differences, mainly Cabernet. Both methodologies of irrigation track each other every week. There's differences, but it's not always on the same direction. So it's not always that the remote sensing is recommending more water or less water. It keeps varying. There's a little bit more different in the Petit Syrah, and we think it's because of the differences also in the actual evapotranspiration value. It's difficult to make conclusions. We only have one Petit Syrah block, but it shows a little bit more difference. When we go to the total water used and the total evapotranspiration of the season, as you can see on the table on the left, for Cabernet Sauvignon, there was practically no difference either in the actual evapotranspiration of the season or in the irrigation applied. And for Petit Syrah, as I said, there were a little bit differences, and we think it's because there's some differences in actual evapotranspiration. And then finally, for block 39, again, there's no difference between both sides of the block. The one that was irrigated based on the remote sensing toolkit versus the one that was irrigated based on the grower expertise. Regarding water status, I'm gonna first focus in the blocks that we added the first year. So the blocks that we had a baseline for 2019, and then in 2020, those same blocks were irrigated based on the remote sensing toolkit. So here you have in 2019 the round symbol, and in 2020 is the triangle. What we wanted to see here is that both years had a similar pattern of seasonal average midday leaf water potential. So we want less stress early in the season, and we want to increase the stress later in the season. I marked a smoke square at the end of the season. I don't know if the results after like August 15 are very reliable, but I still showed them in there. It seems that the remote sensing based irrigation tended to stress a little bit more the blocks early in the season, and this is something we will have to see if it impacts yields or quality, and make sure that it's not something that then has a negative effect over the years. When we go to now 2020, this is only 2020, comparing on the very left side, we're comparing the average of all the control blocks for Petit Syrah and the remote sensing blocks. The blue is remote sensing, the green is control, and we have the average weekly midday leaf water potential. So as I said before, we want to see that the two methodologies track each other so that there's no one irrigation method that significantly stresses the vines more or less at a certain point in the season. And this is what we are seeing here, that the seasonal trends of midday leaf water potential are acceptable, normal, and they are not significantly different between control and remote sensing, except for a point early in the season for Petit Syrah. For block 39, that is the, let's say, the most fair comparison, we don't see absolutely any difference in water status between the north and the south. Regarding yield and quality, I'm going to start first, like I did with the water potential, comparing the baseline for 2019 and then with 2020 when the remote sensing toolkit was applied. In the top right corner, you have the Petit Syrah blocks. This is from the samples that were taken from the vines at harvest. We don't see any significant difference in grape yield in kilos per vine. And then the grape quality index is a measurement that we use at Gallo to select different tiers of grape. Is an internal measurement that integrates a whole panel of aroma and quality parameters of grape. So we didn't see much difference in the grape quality index in Petite Syrah. If something, in 2020 it was a little better. Then for the Cabernet Sauvignon block, similarly to what I said before with the water, the changes seem to be the normal changes between seasons, where some block increase, some block decrease, some block increases, some block decreases, but there's no general pattern that the implementation of remotely sensed irrigation is really having a negative impact in yield and quality. Finally, if we go now to only 2020 and we make the comparisons of the average of the control blocks and the remote sensing blocks for the Cabernet Sauvignon blocks, the Petite Syrah, and for block 39. On the left side, we see the grape yield. And here we see how the control block for Petite Syrah had a bigger yield than the remote sensing block, but there was no significant difference between the Cabernet Sauvignon blocks or between the two sides of block 39. The differences in yield may be because of that difference in stress early in the season, but again, we have one single block of Petite Syrah, so I think we need further data to make conclusions. However, there was no differences in quality. The grape quality index for control and remote sensing was the same in all the cases. And finally, we conclude that the growers evaluated positively the use of the irrigation dashboard as a useful tool for irrigation management, that the remote sensing toolkit was successfully generated weekly over 2 growing seasons by the USDA Hydrology and Remote Sensing Laboratory and delivered to us for real-time use. Also, using the GrapeX Actual Evapotranspiration Toolkit, we succeeded in delivering irrigation recommendations that matched those of a highly experienced vineyard management in Sonoma, California. Regarding each variety, for the Cabernet Sauvignon, the use of remote sensing-based irrigation did not have any negative impact on vine development, water status, or quality. For the Petite Syrah blocks, it seems that the remote sensing-based irrigation significantly decreased the midday liver potential early in the season, and maybe that caused the differences in yield. However, the differences between control and remote sensing may be due to pre-existing differences between block, and we will need to further analyze this variety. And with this, thank you very much. And I don't know if there's time for questions.
The video went black for the past 3-4 minutes or something. So I'm going to see whether the technical staff can rewind so that we can see the end of the video. Okay, in any case, maybe we can go for the questions, right, Nune?
Yes, I think it was, I looked, it was the last 1.5 minutes. I think it was clear anyways. If anyone has questions, a quick question to Mimar, this will be the time. And otherwise, okay, so let's move on. And if we see that this problem is solved, then we still have time and it feels like it makes sense, we can come back to the last part of the video. Now I would like to give the stage to Nicholas Bambach from University of California, Davis. Nico, the floor is yours.
Can you hear me?
Yes.
Okay, all right. So what I'd like to present today is the results from some research trying to use high frequency radiometric canopy temperatures to derive sensitivity heat flux using a new wavelet analysis method. This work has been done in collaboration with a few collaborators from the GrapeX teams, and I've worked really very close with Dr. Macaron to develop this method as well. So the idea here is to come up with a new technique based on wavelet analysis to estimate sensitive heat flux over 30 to 60-minute time period. So resolved at the sub-Daily scale from 1 Hertz canopy infrared radiometric temperature. So it's a very simple sensor that here, it's not my intention to give publicity to any particular company, but the sensors that we've been using is these like radiometric temperature sensors from Apogee that they're very easy to install, and we've had very good results with them. So in particular, all the work that you've been seeing today is based on these sensors. And the idea was to find some reliable and low-cost alternatives to estimate surface fluxes on a continuous fashion. We perceive that while remote sensing tools provide very valuable information resolve at the spatial scale, there is a gap between alternatives that you can continuously monitor surface fluxes, let's say ET, for example, in a sub-Daily scale and in between satellite overpasses. In general, the techniques that we have available to do so, they are costly and fairly complex. So from a commercial standpoint, there are not that many alternatives that they can measure ET in a continuous fashion that can help to ground truth remote sensing products that they are becoming available more frequently for growers. So the theoretical background for this project is based on the idea that temperature in the canopy, while it's dependent on turbulence, there are coherent structures that you can find in the tracers of temperature. And there is a significant amount of research that was done by Dr. Pau and Dr. Gao back in the 80s and early 90s to develop and understand the behavior of coherent structures within canopies. And then from there, they developed a method called surface renewal to measure sensitive heat fluxes at the canopy level based on temperature tracers. During that time, there was a paper from Dr. Pau and collaborators that clearly stated the idea that not only air temperature tracers, but also canopy temperature tracers will exhibit the ramp-like feature or like this coherent structure within the canopy that we could trace, we could measure like if we had like high frequency sensors. Back then, there were not that many alternatives of radiometric temperature sensors that they could measure things at a high frequency, but now we do have sensors that they can do that. What you see in the top slide is like measurements of temperature of air temperature in the dashed line and the continuous line. And the dashed line is like the canopy temperature, and then air temperature is the blue continuous line. And you can see that these ramp-like features can be seen and observed in both in the air temperature and also in the canopy temperature. So we think that there is enough evidence to think that if we can perform some analysis in the coherent structures that we see in the canopy temperature, those are going to resemble what you'll see in the air temperature tracers. The whole idea here behind, if you look at this cartoon, is like if you have air coming from the atmosphere and it goes into the canopy, there is going to be cooler than there within the canopy. It's going to warm up inside the canopy. As it warm up, it's going to get less dense, and then it's going to tend to rise again. And then you're going to have this constant cycle of air coming in and out of the canopy based on temperature and density. And if you trace that, you should observe these like ramp-like features that which is actually what we see in the canopy and the air temperature tracers. So the idea here was to, if we had radiometric temperatures within the canopy, if we could use a wavelet method to reproduce the amplitude and the duration of these ramp-like features in the canopy temperature. So we developed a wavelet net analysis to do so. And what you can see here in black, you have the actual canopy temperatures. In gray, you have the wavelet method filter applied every 15 seconds of frequency. And then you have in red the same wavelet filter method applied to every 60 seconds of frequency. What you can see here at the bottom panel is the IRT temperature wavelet power of spectrum based on the Mexican hat wavelet filter. So at the bottom, you have like a continuous measurements of the canopy temperature for 30-minute period. And then you have the period of each one of those ramps that you observe up here in the top panel. Based on these, we do know that most of the energy within the system, it can be better represented when you go to lower scale times, let's say 15 seconds, which is like the gray line in the picture. And that's basically what we stick to it to our analysis. Most of our analyses are between 15 to 25 or 30 seconds, depending on the canopy that we're analyzing. And we develop an algorithm to optimize which is the best time to capture the frequency of the ramps based on this wavelet analysis. So the whole idea behind this method, it's very simple, and this equation might seem scary, but this is just like your basic physics class and derivation from the enthalpy equation. When you have the sensible heat flux derived from wavelets, it's gonna depend on a calibration coefficient, which is, I'm gonna talk a little bit more about it further in the presentation. The air density, the specific heat capacity, and this is the new component that we add in this equation, which is like a proxy of the amount of energy used to heat up there that it comes into the canopy and then rises once it gets out of the canopy. And basically what we do is like a weighted average of the amplitude and the duration of the ramps that they come into the canopy. So if you look at like an extraction, like a zoom in of one of the ramp events, what we take is the amplitude of this ramp from the wavelet analysis, and also like the duration of the rising time of that ramp. Then that's gonna give you just like the amplitude and the time of that ramp. And also we do it for half of the time to have like a triangle sort of feature that account for the energy in that area, in that time. And then if we do the weighted average, we need to account for the total number of ramps that you have in that total period of time that, let's say, is 30 minutes. So about 1800 seconds, which will be T. And N here is going to represent the number of ramps you have in a given period. And then just to account for the total exchange that you have in the full canopy, then you need to account for the canopy height of that given canopy that you are trying to sample. So basically what we are developing new here is that this abstraction of the weighted average based on wavelet that is slightly different to previous analysis based on surface renewal and using the Vanada approach to characterize ramp-like features in the canopy. So looking at the results, here you have the result for grapevines, but also we did this test for almonds and alfalfa canopies. And you can see that in the y-axis, the wavelets results, these are like 30 minutes fluxes for a period of 30 days. in a vineyard in California, Barelli, which is where Mimar was showing results just now. And then at the x-axis, you see the same result for the flaxes from one of the flax towers. And then that has like all the micrometeorological, very expensive equipment to derive sensitive heat flaxes. You see that there is a fair amount of dispersion between the results, but there is a very good agreement overall across the different canopies. And then when you compare the results also to the conventional sulfur renewal analysis based on the Manada approach for air temperature ramps, also you see that the results are very reasonable. The slope of the relationship, it's within what you can consider like a good method to estimate sensitive heat fluxes, but still there is a fair amount of dispersion that it was worth to take more into like look into the details of that. And when you look at the different canopies, these patterns like sort of hold, but the dispersion is start decreasing as you go in these different canopies. And you see like the results are much more promising in alfalfa, which is the more simple canopy compared to the grapevines, which is a more complex canopy. If you look at the result here in a little bit more detail, you'll see in red surface renewal, in gray the ecovariance, which we consider kind of like the gold standard for our measurements, and then in blue the wavelet method that we developed. On the top panel you have grapevines, in the middle panel you have almonds. Top panel is alfalfa, then almonds, and then grapevines. And what you can see is like the agreement, it's very close for alfalfa, as I said previously, with the exception of some days where it seems like the surface renewal method might show some difference compared to the wavelet, but overall the results are very comparable. But when you go to the grapevine panel, what you see is that there is kind of like a lag in between the peak of the flax of the eddy covariance method compared to the wavelet method and also the surface renewal method. But this gap gets accentuated, especially with the wavelet method. And we think it's because of like the delay time that air eddies can hang out around and within the canopy, that I think they can stay there for longer. And then there's like this like gap that is building through the day of energy that remains in the canopy, and then we say in the late afternoon. So that's something to be aware of. So like our results when we compare, if you look at here, these are results for also on the grapevines using 5 different IRT sensors looking at different aspects of the canopy. And then in the panels, we did the same for almonds and grapevines, and I'm going to focus on results on the results for grapevines. You can see in red the radiometric temperature derived product from wavelets, and in black the results from surface renewal, from eddy covariance, and in gray in the back the derived method using wavelet for the 5 different sensors like looking at the canopy. So you can see that there is a fair amount of dispersion between the different sensors, and then they can have different responses across all of them. And that's going to highly depend on what is the kind of the sensor looking. If there is a sensor looking straight to this, like to the inner row, then you're going to have a flux that is completely different to what you see with eddy covariance. So what you want in this method is to pick that the area that the sensor is looking at is a good representation of the overall canopy that you're trying to sample, because otherwise it's going to introduce a fair amount of bias into the method. So in the one in red that you see there, it's basically the one that we always pick, that is like a depiction of like something that is in between the canopy and between the inner row. And here, to just highlight again the same idea, here you have daily box plot of the fluxes using eddy covariance, surface renewal, and the wavelet method. And then you see like the peak of the fluxes for the wavelet method tend to be later in the afternoon compared to the eddy covariance that is like late early in the morning. And that's because of the delay of heating that we think they might be happening, and then we observe in our system. But when you look at the overall day behavior, we think it's just like kind of like a lag problem. But when you look at the daily scale of the fluxes, you can see that for grapevines, almonds, and alfalfa, the results are much more promising. So at a daily scale, when all the energy gets used in the system, you can see the agreement and the noise. It's better agreement and less noise in the signal. So we move forward using these results, and this is an example for almond in 2019 to estimate ET based in like in an energy value equation. So like deriving ET based on the residual of the energy budget, measuring all linear radiation, sensitive heat flux using the wavelength method, and then ground heat flux using ground heat flux sensors. And then you can see the wavelength method in blue agrees fairly well with the eddy covariant method to the year. There's not much of a difference in the total estimates of ET using our wavelength method compared to eddy covariance, and that's like very promising because it's a much cheaper alternative to measure ET at a daily scale compared to eddy covariance. And also, it's very promising because if you work with surface renewal, these are dependent on frank wire thermocouples that they break very easily. And using the infrared temperature sensor is more reliable because they don't break, and then they can last a long time, and then you can have a more continuous time series of results. So just to wrap up, in conclusion, we think like this new method, it's strongly correlated with eddy covariance and surface renewal method, that they are like considered gold standard for sensitive heat flux estimates. And we think that these developments can ignite the development of like low cost alternatives, sensors that they can serve growers to have continuous measurements of ET at the commercial standpoint. We are also now working with one of our collaborators in Utah State University to use drones and images to apply the same algorithm to the pixel level that we can collect from images, and then derive sensitive heat fluxes using this method from images above the canopy. So you can here see when what Alfonso was doing, our collaborator, when he was launching his drone, and then you can see here the drone like next to the sun above the canopy, and then he was holding that drone for over 15-20 minutes in an area where we had our sample, and then we're trying to compare how those measurements can compare to what we measure with the IRT sensors. So that's my presentation, and thank you for your time, and I'll be happy to answer any question.
Excellent. Thank you very much, Nico. I think we may have time for one quick question. So then we are moving on to the very last presentation of this session, of this evening also. Beth Forrestel from University of California at Davis will give us some insights about very recent conditions given the heat waves in California, maybe opening a window to what we may be having to deal with in the next future. Beth, you're the last speaker of the session. The floor is yours. Is she here? Beth, you are muted. Okay.
Yes.
Okay. Beth?
She just texted me and said she's here.
Okay. Does she have any problems sharing your screen?
Did she join the Zoom?
That's what I'm asking.
Oh, thanks, Andrew.
She's in the waiting room. There you go. Okay.
Okay.
She's in the waiting room. There you go.
Okay,
and I've been trying to share my screen, but it keeps saying it's disabled for me.
Okay, that we need to
In the waiting room. There you go.
Okay.
And I've been trying to share my screen, but it keeps saying it's disabled for me.
Okay, that we need to
In the waiting room. There you go.
And I've been trying to share my screen, but it keeps saying it's disabled for me.
Yeah, we are taking care of it from.
And I've been trying to share my screen, but it keeps saying it's disabled.
Yeah, we are taking care of it from.
And I've been trying to.
Nightmare in Zoom.
I think she might be having problems using Safari.
No,
she's not defined as a co-host, and therefore she can't share the screen. But maybe I try to catch them. Can you send me the presentation maybe quickly, and then I will share it from my end, and you just
sure.
have
Can
to
you hear me? Can everyone hear me?
Yeah.
Okay. Yeah, it just keeps saying I can't share it. Oh wait, now I'm able to. I think you gave me permission.
Okay, good. Oh.
Can you see it?
Yes, right.
Are we good?
Yep.
Okay, thank you so much. And I'm not quite sure what happened there, but I'll move quickly. So I'm going to switch gears a little bit in terms of the work that I've done with many of the people who have already spoken today. And I'm going to talk a little bit more about berry chemistry in response to climate change, specifically in relation to heat waves, in response to heat waves, and how we can use irrigation to cope with that, as that is generally the means that we use to actually mitigate heat wave damage. And so we all know, this is something that should be familiar to people both in Israel and in California, that in Mediterranean regions, we will have issues with both extensive heat, as we will with all over the world, but also drought, and really increasing atmospheric demand and drying soils. So here on the left panel, you see the change in the number of 3-day heat waves. And for the sake of this talk, I realize heat waves are contingent upon the region that you're in. But for what we're going to be talking about, I'm defining it as, in California largely, CalAdapt defines it as 3 or more days where the temperature is above 38 degrees Celsius or 100 Fahrenheit. So 3 consecutive days. And we see that based on relative to the 2080 to 2010 normal, we'll have a massive increase in the number of heat waves across California. And a lot of them will be in the Central Valley. And in addition to that, we'll have increase in VPD and potential evapotranspiration. So increased soil drying and increased transpiration from plants and increased water use. And as Bill mentioned earlier, this is going to be really problematic because we know that there will be, even if there isn't reduced precipitation, we will see a reduction in available water due to shifts in timing of precipitation and lack of snowpack. And so I think it's really relevant in the context of this conference also to mention some of the more recent work that came out about droughts. And we should recognize that both the Western US in terms of soil moisture, not so much precipitation, but in terms of soil moisture and runoff, as well as pointing out Israel, is there'll be a lot more extreme drought risks. And this is going to be really relevant to how we handle water use and heat extremes. And so today I'll be talking about a region that may be less familiar, although it was brought up earlier today, less familiar to some, and this is Lodi, the Lodi AVA in California. Just to orient you, these are a few other regions that my lab works in. This is Napa Valley, just north of the Bay Area, and then this is the Central Coast. And so Lodi is both in Sacramento County and San Joaquin. It's in the San Joaquin Valley, the northern part of the Central Valley. And it actually is one of the most relevant wine regions in California. It has 100,000 acres in wine grape cultivation. It's really this tipping point between some of the wine goes into high quality, or some of the grapes go into high quality wine, into Sonoma and Napa. And there's really a lot of diversity planted there. And if you have heard of Lodi rules, which I believe are starting to be applied in Israel as well, it's really relevant to sustainability efforts. And so that's the location of the study I'll be talking about today. Just to give you an idea of how much warming there will be relative to what there is now in terms of days above 38 degrees C annually. This is just context for California. This is current, so 2015 actually, then 2050 to 2100, and you can see this massive increase again, especially in the Central Valley, of days that are above 38 degrees Celsius. And similarly, this is the Lodi AVA down here, which is located in Central Valley. And then this is the field site. And this is, I should mention, the 8.5 RCP. So if you wanted to take it down to 4.5, if you are more comfortable with the lower threshold of warming potentially and having reduced emissions, you can look at 2050 for what 2100 would be. But either way, there's a pretty dramatic increase going from 0 to a few heat wave or days, 0 to 10 days of heat wave or above 38 C, all the way up to sometimes 50 or 60. So that's a fairly dramatic increase by the end of the century. And if we're going to start thinking about these things in terms of either switching varieties or rootstocks or how we handle this, this is something we need to consider now. And so this study is based on Cabernet Sauvignon. I also work with other cultivars. I've been interested in their using new cultivars to actually cope with climate change, But I think we have to recognize that both in Israel and in California and many regions of the world, actually the most widely planted variety in the world is Cabernet Sauvignon. And I hope that many of you recognize Margalit. He actually has a lot of relevance in Davis as well. He's written some books and was here on sabbatical several years ago. And I had the great opportunity recently to also try some other Israeli wines, which we're actually working to bring a few of them over. Baladi and Debuki actually make really exceptional wines, as do some of the whites. And so I would be remiss not to mention those, because I think they're really interesting and have a lot of capacity for heat and drought adaptation. As far as this project goes, this is something that was largely carried out by a master's student in my lab, who is co-mentored by Andrew McElroan, Martina Galliano. She was a winemaker at Terpenes and has become a phenomenal viticulturalist. And this is also in collaboration with Ian J. Gallo, who really made this study possible. And so in Lodi, in the Lodi AVA, there is a variable rate drip irrigation system that was started by Ian J. Gallo. Again, it's Cabernet Sauvignon on 1103P rootstock. It's mechanized pruning, leafing harvest, high wire trellis. So there is some shading there. And there's, as I said, the variable rate drip irrigation system, which we took advantage of. And it ended up being these 30 by 30 meter square pixels. And what we did was select replicates of these across this fairly heterogeneous vineyard. So there was quite a bit of topographical and edaphic diversity within this site. And what we did is we only manipulated irrigation during heat waves. And this is really important. So growers obviously can't make a decision in real time for heat waves. And we conducted a survey assessing across California what growers actually do in terms of heat waves. And they largely, over 90% of them, if it is 100 degrees or more, or 38 degrees Celsius or more, they will ramp up irrigation prior to a heat wave. And oftentimes, I think there's about 60% of them that continue that irrigation into the heat wave. So you see a heat wave coming based on the forecast, and you often will put on a decent amount of water. And I was actually surprised to find that it was quite a bit of water that people would often use, that growers would often use. So the way that we designed our treatments is by having a baseline treatment. So 1 to 2 days prior, and then into the heat wave, we applied the deficit irrigation of 60% ET, which was estimated by remote sensing techniques established by Gallo. And then the other treatments were 2 times that baseline before and during the heat wave, and then 3 times that baseline. And so I'm going to present to you 2019 data first, and then just give you a little bit of our preliminary data from 2020. This has been ongoing for 2 years, 2 seasons now. And so in 2019, there were 2 heat waves, one right before veraison, and then one later in the season afterwards in August. And you can see here, there's the baseline. All of the background irrigation was determined by trying to standardize the NDVI. So there aren't huge changes in the background irrigation, just slight variation. But where we had massive changes was during and prior to these heat waves, where you can see the baseline, 2 times that, and 3 times that. And this color scheme will be used throughout to refer to the baseline treatment, the 2X and the 3X. But just remember that it's only application during these 2 heat waves that changed. So when we look at stem water, midday stem water potentials, which is largely used to assess stress, it was interesting because actually prior to the first heat wave in pre-veraison were some of the most negative stem water potentials. And there was a decent amount of water application mid-season. So you don't really see much of an effect until after heat wave 1. So you see the baseline where it was maintained deficit irrigation, and then after heat wave 2, but we always saw recovery. And you should note that there was always recovery by the week after. In this case, we don't have another sampling point because there was a heat wave so soon afterwards, but by the time harvest came up, there was full recovery. As far as the physiological responses, this is during heat wave 2, during the second heat wave. And you can see that net photosynthesis declined and the leaf temperature reached higher temperatures. This is showing Liquor leaf temperature. We also did IRTs, which are the same trends. We used IRTs to look at temperature as well. I'm showing the Liquor leaf temperature here based on the gas exchange system because we had more data points. But you can see there's this depressed net photosynthesis due to the heat wave and largely depressed stomatal conductance. There is no significant difference between the 2x and 3x for these physiological responses. And so, again, this is actually looking at the actual yield, not the leaf temperature and gas exchange anymore. Sorry about that. But you can see that the result of this reduced photosynthetic rate and stomatal conductance and some of these responses just during the heat wave, again, resulted in a reduced yield for the baseline, a significantly reduced yield for the baseline, both in terms of kilograms per vine and grams per cluster or grapes per cluster. So as far as chemistry goes, we also saw during the 2019 season that there is a more rapid maturation and a more rapid accumulation of bricks in the baseline. And so we didn't actually see any sort of cessation in sugar accumulation, which you sometimes do see in heat waves. These heat waves were perhaps not extreme enough, but we did see that due to those heat waves in the baseline, we had a more rapid accumulation. Our target harvest was 25 to 26 bricks, which is why you see this stopping here, because the 2x and 3x reached those later. So we did see higher bricks. We saw a significantly higher pH and significantly lower TA. And then what I think was one of the most interesting results is that we found that there was a reduction in tannin, and we don't know if it was synthesis or degradation. We're trying to tease that apart now. But that first heat wave that was prior to veraison, so tannin synthesis begins in flowering. It's often finished by the end of veraison. And so what we did is we saw this heat wave effect from the first, just in having 3 days of differential irrigation that was maintained throughout the second heat wave and resulted in a significant decrease in tannin per berry in the baseline. And what was really interesting too is that there was this negative effect of overwatering as well. So there's really this balance, which makes sense. You maintain deficit irrigation to acquire the phenolic compounds and the concentration you want. But I don't know that we recognize how relevant that is when we think about watering for heat waves, when it's just a very short event that we're increasing the water and the irrigation. So that was an interesting finding. Again, there's this negative effect of not providing irrigation, excess irrigation in terms of anthocyanins as well. And again, you see that 2 times baseline being the highest amount of anthocyanin and the negative effect of applying excess water. And I just want to mention too, we did dive into some of the monomeric anthocyanins and some of the other compounds. And it's worth mentioning that the malvidins were what were driving most of this pattern, which is really related to color in berries. And we won't talk about the wine chemistry, or I won't talk about the wine chemistry in this talk, largely because we only have 1 sample point, which was at bottling from the 2019 season, and we weren't able to do sensory or subsequent analysis. So they were in my lab, now they're sitting at in the wine cellar here. And so eventually, fingers crossed, they'll be very well aged. We'll hopefully be able to do sensory study. But it was really interesting to find that this was the primary compound driving the results as far as anthocyanins. So I'm going to quickly move into our 2020 field season, because 2019 was it was right before I started my professorship, and it was kind of a crazy time even being able to get out there and do all those diurnal measurements. We did a ton and did a ton of pre-dawns. And so our goal was to get these micromet stations out there. And this was in collaboration with Nico Baumbach and the Macaron Lab and others, Brian Bailey and Plant Sciences. And so our hope was that we could get these out there and monitor throughout the season. Unfortunately, COVID hit, and so we were delayed slightly, more than slightly, but we did get them out there for the second set of heat waves, and we're working on analyzing that data. But I think a few things to mention about this data set, which we're still analyzing as we just finished harvest in September. We expanded the 30 by 30 meter grid cell or pixel to 60 by 60, so we could have a greater imprint and actually look at ET and do some surface renewal and use the wavelet, eventually use the wavelet method that was just previously discussed. And these should be filled in down here. I'm not sure why they're not. But also, as I said, we had all these other sensors out there, and we also monitored the berry temperature, and we're able to do some remote sensing and some hyperspectral measurements. And as many of you know, 2020 ended up will likely be the warmest year on record. And so we had 4 heat wave events, and it was a very intense season. Heat wave 3 was exceptionally long. This is also when the fires occurred in California, which between COVID, fires, and all else, it was quite a special season. So I'm going to focus a little bit on the results from heat wave 3 and 4, as that was post-Veracian and what we've analyzed so far. And so again, you can see the same trend we saw from 2019 in terms of the heat wave was September 5th through 7th, and you saw this reduction in stem water potential and reduction in stomatal conductance, but then you saw recovery within 5 to 7 days by the time we harvested. Similarly, you saw a reduction in net photosynthesis and recovery, even in the baseline treatment. And then we did see, even though there's much more variation because we had many more pixels and there's a lot of heterogeneity, we did see a significant lower yield, again, in that treatment, in the baseline treatment. The berry chemistry was a little bit more complex, and we're still working with that. Interestingly, we did see some reduction in tannin, but there seemed to be a recovery after heat wave 3, which is interesting. And we have more time points in here that we're currently analyzing, but it's interesting to see that either there was some degradation happening in the other treatments or in the baseline treatment in the 2X and 3X treatment, because usually synthesis is done occurring by then, but we'll be able to tease that apart with our other data. But again, in heat wave 3, we saw, again, recovery for anthocyanins. Sorry, this is the Spanish version of anthocyanins, I think. A recovery, but then heat wave 4, again, reduced it for the baseline. So we're seeing very similar patterns, but a little bit more complex. So what I want to touch on for the end of my talk, especially since it relates to some of the previous discussions in the next session, is really the potential for remote sensing of heat stress. And so between the Open ET project, which was discussed earlier and will be released soon, EcoStress, which is thermal data from Jet Propulsion Laboratory, and we have collaborators there. And then this EcoStress is a satellite, but Avarice will, the hyperspec spectrophotometer on Avarice, which is an airborne spectrophotometer, will become eventually a satellite in the years to come. And we actually had this great opportunity to have them fly over the heat wave site and collect hyperspectral data, so 400 to 2600 nanometers. Unfortunately, that data is extremely difficult to analyze because of the VOCs and the fact that there were raging fires across California at that time. So we're working with that, but we also had the opportunity to work with the Magni Lab and take leaf level and canopy level samples. And I'm just going to show you very briefly a singular value decomposition, basically a PCA of the spectral data. And this is from all the samples, and this is from the third and fourth heat waves combined. In this case, treatment 1 is the baseline, treatment 2 is 2x, treatment 3 is 3x. And so I'm going to show you just the SVD for the 400 to 900 nanometers of wavelength, because that was where there's the most variation. And you can see that we have this portion really stand out as being different for PC2. And there's also a relationship to non-photochemical quenching from our physiological data between those treatments as well. And this should be 1, 2, and 3. And this suggests that there really was, there's increased absorption in the xanthophyll pigment or carotenoids, which has related to photoprotection in the spectrum. And so I think this shows promise in the ability to potentially use some of these remote sensing techniques. And we know too from others' work that some of the thermal imagery, such as ecostress and otherwise, might actually be indicators before you see some of the physiological stress occurring that may help in terms of water application and an active response. So as far as our studies moving forward, as I said, that data was really just preliminary for this past season. And we're going to be pulling all of the micrometeorological data from the canopy and from the berries. We monitor berry temperature and other parameters, and we're going to link those to the physiological and chemical responses. Due to all these heat waves, we had 10 sampling points plus harvest, so there's a ton to process for the berry physiology. We also have data to analyze molecular responses, so it'll be really interesting to see whether you're seeing promotion or you're seeing synthesis changes or degradation in some of these parameters. And then lastly, in collaboration with Bill Kustis and others, the goal is to expand this to other sites and other cultivars so that we can integrate more remote sensing efforts in some of this work. And so there are many, many people to thank and acknowledge, and these are our many collaborators and funding agencies. And again, I especially like to thank E&J Gallo for their in-kind support and all of their support throughout this project. It would not have been possible without them. And so thank you. We did maintain social distancing while sampling this past season. And thank you for your patience with me as well as I got that figured out.
Thank you very much, Beth. It's quite worrisome and very interesting. If there is any question to Beth, please go ahead. Okay, no questions. Well, this weird platform of online conferences, I'm still figuring it out. And mostly dislike it, I must say. And I really hope to have all of you next year in a conference like this in Israel. We'll see how it goes, and we'll see what the future brings to everybody. I want to wrap this session with thanking all our speakers for both the earlier and this later session. Special thank you to those of you who had to wake up rather early to join us on time. Another challenge of this weird platform. And I want to remind everybody of the session that we're going to have on Wednesday at 4 p.m. Israel time. That will be a continuation and upscaling of what we've heard today. And if no one has any further comments, I would wrap this session. Aaron, do you want to say anything?
Yeah, I also would like to thank you very much, because I was completely unsure how this would go, and it went almost smoothly. So Again, thank you for your patience and for your participation. And see you in Israel or California or Canada or wherever, just out of here.
Thank you very much. Bye-bye.